Good evening, students. So this is my fifth video when it comes to NEET revision. As you have told you earlier also, I am preparing NEET revision videos. Remember, these are revision videos, not direct detailed conceptual videos. From these videos, you will be getting direct questions for your next NEET examination. I'm preparing these videos for a re-NEET exam that is to be conducted on June 21. So this one video can give you more than 30 marks guaranteed, and all the content will be exam orientation, high yielding and integrated with the previous year questions. Now moving on, in this video we are going to cover three chapters actually: biological classification, plant kingdom and animal kingdom, and minimum of like 8 marks are being asked from all these three chapters combined, and probably you will be scoring nearly 30 marks if you just focus this video and also prepare this video, stop the video and revise multiple times. Okay, first you have to watch the video in one time in a normal speed, and then you have to watch the whole video 2 to 3 times in 2x speed, and that's how it will help you. Examiner's blueprint, like from biological classification, what topics have been being expected and what was the previous pattern. So when it comes to kingdom Monera, kingdom Protista and kingdom Fungi, and these viruses, Viroids, Viroids and Prions and Lichens, high weightage has been expected. So out of 8 questions, like every two questions from each kingdom, and or 3 questions sometimes have been directly asked, and type of questions also I have given here to just to give you an idea. Obviously this is a blueprint, but obviously he can ask more questions or lesser questions, it depends upon the examiner. But before going into the topic directly, we are going to see that what topics have been expected in the previous year questions. Let's see about the classification. What do you mean by classification? Why should there be a need for classification? Why do you have to classify the organisms in one category to another category? That is a concept here we are going to study. Okay, so need for classification and historical background. Let's see early classification systems. Aristotle was the earliest scientific classification. It was given by Aristotle. Classified plants into trees, shrubs, and herbs based on simple morphological characters. So the basis of Aristotle classification was simple morphological characters. Divided animals into those with red blood, that is vertebrates, and those without, that is invertebrates. But this classification was scientifically wrong. It was proven later on. Now comes the Linnaeus. Antony von Linnaeus, like he gave two kingdom system, that is Plantae and Animalia. This system did not distinguish between like eukaryotes and prokaryotes, unicellular and multicellular. He just gave photosynthetic, that is green algae, and non-photosynthetic. This also definition was not present. He only gave plant and animal differentiation. That is called as two kingdom system given by Linnaeus. The drawbacks of this two kingdom system is a large number of organisms did not fall into either category, that is example bacteria, fungi, protozoa. Gross morphology alone was insufficient. So that was the drawback of the two kingdom classification given by Linnaeus. Then comes the R.H. Whitaker's five kingdom classification given in 1969. So Whitaker gave five kingdom classification, and the main criteria used by the Whitaker in order to give the five kingdom classification is cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. So cell structure, the structure of a cell, presence and absence of cell wall, cell membrane, and the functions of special cell organelles, all these was taken under the criteria. Body organization, is it a cellular level of organization or tissue level of organization or organ system? That criteria was also undertaken. Mode of nutrition, either it is a autotrophic or heterotrophic or saprotrophic or chemoautotrophic, etc. And also basing on the reproduction, like either it is asexual reproduction or asexual. In asexual also, what is the type? And phylogenetic relationships, it means relationships in the evolutionary tree. These all were the criteria used by Whitaker, and this could be a direct question. What was the criteria, or which among the following is not the criteria of Whitaker's classification, could be a question. So the five kingdoms were Monera, Protista, Fungi, Plantae, Animalia. So these were the five kingdoms of the Whittaker's classification. Let's see one by one. Monera. Monera are prokaryotes, bacteria, cyanobacteria, mycoplasma. All these come under the kingdom Monera. Protista are unicellular eukaryotes and fungi are multicellular heterotrophs with chitin cell wall, and the plantae, as you all know, are multicellular autotrophs cellulose cell wall, and animalia are multicellular heterotrophs and no cell wall. True or false questions or statement based questions with inverse integration could be asked from these statements. This system was a major advancement as it considered fundamental biological criteria and evolutionary relationships among the organisms. So stop the video here and study the previous year questions, how the questions have been asked from this topic, so that you can integrate your topic also and you'll come to know the examiner's mindset. Moving on, let's see each and every kingdom in detail. Kingdom Monera. The general characteristics of kingdom Monera being it a prokaryotic, it means no true nucleus. It means no membrane bound organelles. The nucleus and some organelles don't have the membrane of their own. Such type of cellular structure is called as prokaryotic cell. Cell wall. Non-cellulosic, it means polysaccharide plus amino acid peptidoglycan. That is the cell wall characteristic of the Monera. When it comes to body organization, it is cellular, mostly unicellular, colonial or filamentous, and mode of nutrition autotrophic, photosynthetic or chemosynthetic or heterotrophic or saprophytic or parasitic. All such type of things can be expected from these mode of nutrition in the Monera. And bacteria are the sole members of the Monera. They occur almost everywhere. Extreme habitats, hot springs, deserts, snow, deep oceans also can be seen in the habitats can be seen when it comes to Monera. Now let's see, based on the shape, the bacteria. Bacterial shapes, there are four types. Spherical shaped bacteria are called as coccus or cocci in a plural term. Rod shaped is bacillus or bacilli, comma shaped vibrium or vibriae, and spiral, that is spirillum or spirilla. So metabolic diversity also seen. This is NCERT important topic when it comes to the metabolic diversity of the Monera. Autotrophic bacteria and heterotrophic bacteria. In the autotrophic bacteria also, there are two types. One is photosynthetic autotrophic and chemosynthetic autotrophic. The photosynthetic autotrophic example are cyanobacteria, blue-green algae, have chlorophyll A and perform the oxygenic photosynthesis, whereas in the chemosynthetic autotrophic bacteria, they oxidize the inorganic substances like nitrates, nitrites, ammonia, iron, sulfur to produce ATP. They play a great role in nutrient recycling. So a role played on the nutrient recycling are the type of bacteria in which the metabolic activity is chemosynthetic autotrophic. Now comes the heterotrophic bacteria. Most abundant are these heterotrophic. Example, decomposers break down dead organic matter, helpful in nutrient cycling, and they are helpful in curd formation, that is lactic acid bacteria, antibiotic production. Example, streptomyces, nitrogen fixation in the legume roots, that is rhizobium. The relationship between these rhizobium bacteria and the leguminous plants root nodules is symbiotic in nature. Pathogens cause diseases like cholera, typhoid, tetanus, and citrus canker. You can see there are two previous questions I have given here so that you can integrate the topic with the questions. Now comes the archaebacteria. Ancient bacteria. Key points about these archaebacteria: live in harsh habitats like Monera also. Halophiles. Halo means salty areas, and phile means liking. Phile means like. If I like salt, then I'm also a halophile. Phili, it comes from phili. Halophiles, extreme salty areas, salt pans, salt beds. Thermoacidophiles, hot springs, high temperature, acidic pH. Methanogens, marshy areas, gut of ruminant animals, cows and buffaloes produce methane biogas. So halophiles, thermoacidophiles and methanogens all come under the archaebacteria. Direct statement based and fact based questions can be asked from halophiles and thermoacidophiles and methanogens, because these bacteria have their own special character, and also the name is coming from that character only. Differ from other bacteria in the cell wall structure, but it means there is no peptidoglycan. They have only unique lipids. You can see in the diagram here. The cell wall difference is other bacteria has peptidoglycan, whereas in the archaea bacteria there is no peptidoglycan and unique lipid composition. Ether linked lipids are present. Methanogens are responsible for methane production from dung. So archaebacteria are primitive prokaryotes adapted to extreme conditions and play important ecological roles. The types and habitats, the name itself tells you. Halophiles live in extreme salty conditions like salt pans and salt beds. Thermoacidophiles live in hot springs like high temperature and acidic pH. Methanogens live in marshy areas, gut of the ruminant animals, cows and buffaloes, and produce methane biogas. You can stop the video here and study the previous year question. Kingdom Monera. Monera is actually eubacteria, true bacteria, rigid cell wall, if motile, have flagellum also. Cyanobacteria, that is blue-green algae, have a chlorophyll A. They are photosynthetic autotrophs. They are unicellular or maybe colonial or filamentous, often form blooms in the polluted water. Some have heterocysts. What do you mean by heterocysts? Heterocysts are specialized cells for nitrogen fixation. So nitrogen fixation is very important actually. We are going to study the nitrogen fixing bacteria also, but here in the cyanobacteria, heterocyst is the specialized cell that is present along with the normal vegetative cell that are going to do the examples of this nitrogen fixation bacteria are nostoc and anabina. So they come under the cyanobacteria. The blue-green algae is actually a misnomer. It is not properly algae. The heterocyst is thick-walled and provides the anaerobic environment for the nitrogenase. So this heterocyst, the specialized cell, is a thick-walled cell, and it is going to provide the anaerobic environment for the nitrogenase. The diagram of the nostoc and anabina is also important. Diagram based questions directly can be asked. This question about kingdom Monera has been asked previously also. Stop the video here and study the previous year question, that will be helpful for you. Moving on, let's see with the reproduction in bacteria. Mainly the reproduction in bacteria is asexual. Main reproduction, most common and most frequent reproduction that is happening in the bacteria is asexual, and that too it is a binary fission. What is a binary fission? Let's see. Okay, a parent cell, direct there happens a DNA replication and septum formation within a parent cell, and that's how it divides into two daughter cells. So one parent cell, one DNA divides into two DNA, septum formation, one cell into two cell. That is called as binary. Bi means two and fission means breakdown. So binary fission is a asexual reproduction and the most common reproduction in the bacteria. Under unfavorable conditions, it means those conditions which are not suitable, which are bad, which are not favorable. Under those conditions, they produced by endospores. So what is endospore formation? Again, when we say a normal cell, that cell under the unfavorable condition converts into a spore formation. The spore is internal, and that when it matures, it becomes the endospore, and then the spore is released outside after the breakdown. Lysis means breakdown. So lysis means breakdown. That is endospore formation. Now when it comes to sexual reproduction, the primitive DNA transfer takes place by three processes. One is conjugation, two transformation, and three transduction. So first of all, by heart the three types of sexual reproduction happening in the bacteria, that is conjugation, two transformation, 3 transduction. Now among all these three, what is the main difference actually? Let's see the difference. In the conjugation, the primitive DNA transfer takes place through sex pili, a type of pilus. Okay, you can see here this is the pili, and from donor to there is a recipient, the transfer of DNA takes place, and that's how it is also considered to be a sexual reproduction. In transformation, it happens. What happens here? Direct uptake of the DNA, a free DNA from the environment is happening. It is called as transformation. And in the transduction, these transfer of the DNA happens through bacteriophage. Bacteriophage transfers the DNA to another bacterium. Now here is the previous year question. Stop the video here and study this previous year question so that you can integrate this topic with the previous year question, and so that you can also understand what could be the further expected questions from this topic. Now comes the mycoplasma. Okay, now mycoplasma has a special categorization because of few features. Let's see what are those. Mycoplasma completely lack a cell wall. Smallest living cells known as zero point 0.3 micrometers and can survive without oxygen. That is, they happen to perform a special type that is called as facultative anaerobes, and they are pathogenic to plants and animals, also called as PPLO, that is pleuropneumonia like organisms. So the another name of the mycoplasma is PPLO, that is pleuropneumonia like organisms, and they are pathogenic. They can cause diseases in both the plants and animals. They can live without oxygen, called as facultative anaerobes, and they completely lack a cell wall. You can see the structure of the mycoplasma also. The key facts are they are very small in size and they lack a cell wall, but obviously the plasma membrane is present. Now you can see the previous year questions also and analyze how questions are going to come in the examination. Now comes the kingdom Protista. The general characteristics of the kingdom Protista are they are unicellular eukaryotes. Now previously we have studied all about prokaryotes. Now we are studying about the eukaryotes. It means they have well-defined nucleus and membrane-bound organelles. They are primarily aquatic and reproduce asexually through binary fission, budding, etc., and sexually through cell fusion and zygote formation. Link with plants, animals, and fungi. It means they are having evolutionary link, a connective link with both plants and animals and fungi. Groups included in these Protista are chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. We are going to study all these groups individually in detail. Let's observe the general characters now. You should remember that protistans are eukaryotes. They have well-defined nucleus and membrane-bound organelles. They are primarily aquatic. They reproduce asexually and sexually. So diversity in Protista, you can see they are having groups like chrysophytes, also called as diatoms, dinoflagellates, euglenoids, slime molds, and protozoans. Let's go one by one. Chrysophytes. Chrysophytes are basically diatoms. They are also called as diatoms, and they are golden algae desmids. Okay, so whenever you hear the word desmids, you hear the word golden algae, and when you hear the word diatoms, you should remember that they are a class of protistans called as chrysophytes. So cell walls. The cell walls of two overlapping silica impregnated shells fit like a soap box. You have to see the diagram here. Okay, these silica shells, they fit onto each other like a soap box. You can see the diagram. It is very easy to understand once you picturize it. That cell walls are called as frustules, and walls are indestructible because they are made up of silica. Because they are made up of silica, these walls cannot be broken down at any cost. So that is the thing here. And moving on, Statement based questions are going to come from this topic, and this could be confusing you when you are going to study the other kingdoms also. So before that one, let's be very clear first of all. Okay, diatoms are basically protistans. Diatoms are unicellular eukaryotes. They have well defined nucleus and membrane organelles. They are primarily aquatic and they are linked with plants, animals and fungi. The cell wall is basically frustules. It means it is made up of silica, that is two layer silica impregnated shells that are overlapping on each other like a soap box. You have to remember this one at any cost. You cannot forget this one. Okay, and the walls are indestructible. You cannot break down the walls because of the presence of these silica. Accumulate over billions of years, forming a diatomaceous earth that is gritty, used in polishing, filtration of oil and syrups. Diatoms are chief producers in oceans, and they are microscopic and they flow passively called plankton. So you have to remember these characteristic features when it comes to chrysophytes. Now, one more important key point is diatoms are important for ecology. Major primary producers in oceans and economics, diatomaceous earth is an industrial use. So, both ecologically important diatoms and economically important diatoms. What is the ecological role of the diatoms? Is basically they are primary producers in the oceans. That is the ecological importance. Now, when it comes to Protista, let's see the other group, that is dinoflagellates. Okay, one group we have studied is chrysophytes, the other group is dinoflagellates. So, dinoflagellates are mostly marine and photosynthetic. Cell wall is made up of stiff cellulose plates like an armor, like a soldier wears her armor, like a shield type structure. Now, that is called as armor. So, the cellulose is so stiff that it is looking like an armor around that cell. That is the characteristic feature of the cell wall, or armor like or cells stiff cellulose plates are present in dinoflagellates. The pigments are yellow, green, blue, brown, red depending on the main pigments. There are two flagella in these organisms, that is the reason they are called as di and flagellates. The two flagella, that is one is longitudinal and one is transfer in a furrow. The red tide, this is very important. Due to rapid multiplication of red dinoflagellate, example, uh, goniolax, toxins released kill marine animals, and this is called as the red tide. This happens in the sea sometimes. Okay, now moving on, euglenoids. The third category is euglenoids when it comes to protistans. They are mostly freshwater. So dinoflagellates are marine and euglenoids are freshwater, and euglenoids habitat under the stagnant water. There is no cell wall, have pellicle, that is protein rich layer, that is flexible body, and two flagella are present, one short and one long. Even in the dinoflagellates also there are two flagella, and in the euglenoids also there are two flagella, but here the flagella are one longitudinal and one transverse, like they are like perpendicular to each other, whereas here it is one short and one long. Here in dinoflagellate there is a cell wall, stiff cellulose plates are present, where here in euglenoid there are no cell wall, and they are mixotrophic. It means they are photosynthetic in sunlight, pigments identical to higher plants like chlorophyll A and B, but in darkness they behave as heterotrophs, predate on smaller organisms. Example is euglena. You have to even remember the diagram of the euglena also. It has one long flagellum, one short flagellum. There is an eye spot. There are chloroplasts in this one, and it contains chlorophyll A and B, and there is a nucleus and a pellicle, that is, it doesn't have a cell wall. In sunlight, these euglenoids behave photosynthetic, and in darkness, they behave as heterotrophic. The key point you have to remember is dinoflagellates are marine, cellulose, armor present, and cause red tides, whereas euglenoids are freshwater, no cell wall, and mixotrophic, that is, switching mode of nutrition is seen in the euglenoids. Stop the video here and study the previous year question. There are match the following statement based questions also, and these questions can come directly again. And basing on that previous year questions, try to understand this slide, and we are going to cover the expected questions also in the further slides. Now comes the slime molds. Slime molds are saprophytic, grow on decaying twigs and leaves and engulfing the organic matter. Plasmodium and aggregation of slime molds under favorable conditions. It is a multinucleate mass that spreads and under the favorable conditions form the fruiting bodies bearing spores. True walls, they are resistant, survive for years and spores dispersed by air currents. So this is the characteristics of slime molds. Slime molds show both unicellular amoeboid and multicellular plasmoidal characteristics. Common genera are Physarum, Fuligo, and Dictyostelium. So you have to repeat after me and by heart now itself. The common genera that comes under the slime molds are Physarum, Fuligo, and Dictyostelium. So the life cycle of slime molds, they have like first plasmodium stage, that is multinucleate mass, spreads and feeds. Then comes the fruiting body formation under unfavorable conditions. Then there is a spore release, spores dispersed by air currents. Then comes the spores germinate under the favorable conditions to form amoebae. So amoeboid also and the plasmoid of the lifestyle also seen in the slime molds. This topic is very important because the behavior of the slime mold is somewhat unique and characteristics. The key points you have to remember is they are saprophytic organism. The plasmoidal stage is multinucleate and form fruiting bodies with spores under the unfavorable conditions, and spores have true walls and are highly resistant. Example, Physarum, also called as dog's vomit fungus. The common name for the Physarum slime mold is dog's vomit fungus. Stop the video here and study the PYQs. Kingdom fungi. They are heterotrophic, eukaryotic, and they are non-vascular. The cell wall is composed of chitin or chitin, that is, it is made up of polysaccharide and polysaccharides. Body is filamentous except in yeast, which are unicellular. Filaments are hyphae, and the network of that is called as mycelium. Hyphae may be xenocytic, it means aseptate, there are no cross walls and they are multinucleated, or hyphae may be septate, that is with cross walls, there are septa and they are having pores. So xenocytic and aseptate and septate hypha are characteristics of fungi. When it comes to nutrition, they are saprophytic. It means they absorb the soluble organic matter from dead substrates. And they are parasitics also, that is, they are living on living plants and animals. And they are symbiotic as seen in lichens with algae and mycorrhiza with roots of the higher plants. When it comes to reproduction, there are four things we have to understand. One is fungi goes into reproduction through vegetative process, that is fragmentation, fission, and budding. What do you mean by fragmentation? It is like breaking down of one hyphae and gives rise to the each broken part converts into a original fungus. Second is fission. It means one cell breaks into two, and budding, it means from one cell itself the other cell comes out of it, it pops out, that is called as budding. Asexual type are through conidia, those are exogenous, and sporangiospores, which are endogenous, and zoospores, which are motile. So exogenous are conidia, those spores are spread outside, that those are exogenous, and sporangiospores, in which the spores are present inside endospores, and that is what we have to understand about the third difference, that is zoospores. It means they are motile, they have flagella like structures also, you can see in the diagram. Sexual reproduction involves plasmogamy, where the fusion of protoplasms, karyogamy, fusion of nuclei, and meiosis, in which the zygote producing haploid spores are seen here. You can see the diagram. In the plasmogamy, there are the fusion of protoplasms, and the dikaryotic stage n plus n converts into a karyogamy, that is, there is a fusion of nuclei, and that goes into meiosis in zygote, and the haploid spores are produced in this reproduction, that is specifically seen in fungi. Fungi play crucial role as the decomposers and parasites and mutualistic partners in ecosystems and have immense economic importance when it comes to food, antibiotics and biochemical industries. Stop the video here and study the previous year questions. These previous year questions will tell you that what type of questions have been asked previously, and when integrating that PYQs with the slide you can understand what could be the future expected questions in the NEET. The key takeaways are they are heterotrophic eukaryotes with chitinous cell wall. You should remember that chitin is made up of polysaccharides, and they are filamentous body, that they are hyphae, and that hyphae can also be converted into mycelium except in yeast, because yeast is a unicellular fungus. Hyphae can be of two types, one is xenocytic and another is septate. Reproduce vegetatively, sexually and asexually also, and they are showing saprophytic, parasitic and symbiotic nutrition. Moving on to Phycomycetes. Okay, this category are also called as algal fungi. You should not confuse either it is a algae or it's a fungi. Basically, it is a fungi, but it has some features of algal appearance only. That is why they are called as algal fungi. We'll see here. The habitat of Phycomycetes, they are aquatic, decaying wood in moist places and obligate parasites on the plants. So they are found in three areas. One, purely aquatic, that is in water. Second, they are present in the decaying wood in the moist places. It means there should be some moisture, there should be some water droplets in that area, and that type of environment, if the wood is decaying, then these Phycomycetes fungi can go there and grow, and they are obligated, means compulsory parasites in some of the plants. They have mycelium, that is both aseptate and cyanocytic. The asexual reproduction happens by zoospores, as we have studied in the previous slide that the zoospores are motile, or they are also showing some aplanospores, that is non-motile spores, that go into asexual reproduction and produced endogenously in sporangia. Sexual reproduction by zygospore formed by the fusion of the gametes, that is isogametes, similarly looking gametes, and anisogametes, which are dissimilarly looking gametes, or oogametes, where they are going to fuse with the female and male parts. That is called as sexual reproduction. The examples are mucor. Mucor is a common bread mold also. Rhizopus and albugo parasite on the mustard causes the white rust. These examples are very important. Direct match the following or exam based question can be formed from here. You can see in the diagram in the asexual reproduction the zoospores are motile. They are being burst out of the sporangia. They spread and form their own life, and aplanospores are non-motile. And when it comes to zygospore formation, you can see there is a formation of gametes, then gametes are being fused, then there is a formation of zygospore which is thick walled. And when it comes to examples, you have to remember this mucus and mucor and rhizopus and albugo on the mustard. Stop the video here and study the previous year question. The key points you have to remember about the Phycomycetes: they are aseptate, that is they are cyanocytic. Mycelium reproduce asexually by zoospores or aplanospores in sporangia, and sexually by zygospores. Examples are mucor bread mold and rhizopus and albugo, that is white rust of the mustard. Now comes the Ascomycetes. Ascomycetes is also called as sac fungi. Why they are called as sac fungi, we are going to see, because the sexual reproduction happens in a sac-like structure called as asci, and that is why they are called as Ascomycetes, that is sac-like structure. The habitat is they are saprophytic, decomposers, parasitic and coprophilous. It means they are going to eat the dung. So the mycelium is septate and branched, and asexual reproduction happens by conidia exogenous produced on conidophores. The sexual reproduction produce ascophores endogenous inside sac-like asci, singular ascus, and asci are arranged in fruiting bodies called ascocarps. So in these fruiting bodies called as ascocarps, they are sac-like structures called as asci, in which they are ascospores, they are endogenous spores through which the sexual reproduction happens. That is why this type of fungus is called as ascomycetes. You should remember the examples, especially the examples are Penicillium, Aspergillus, Claviceps, Neurospora, Saccharomyces, and Morels and Truffles. When it comes to Penicillium, it is a source of penicillin, a common antibiotic also, and this penicillium is commonly found on the bread and fruits. This is a fungus basically. Aspergillus produces citric acid. Some species causes diseases also, and Claviceps, they are parasitics on grasses, source of alkaloids and LSD, and Neurospora used in biochemical and genetics studies, and Saccharomyces yeast unicellular used in fermentation, and morels and truffles are edible, they are delicacies. When it comes to yeast, you have to remember that it is a unicellular ascomycetes, also an example of Saccharomyces. The reproduction is by budding vegetative and also forms the ascospores. Ascomycetes are ecologically important as decomposers, and they are economically useful in food, fermentation, production of antibiotics, and organic and acid production. Stop the video here and study the previous year question. You can see what type of questions have been asked in the previous year in the NEET examination, and after studying the previous year question, you can integrate it with the slide and understand how further expected questions can come out of it. And the key takeaways are asci contain ascospores endogenous, and conidia are exogenous asexual spores, and ascocarps protect and aid in spore dispersal, and yeast is unicellular. Other ascomycetes are filamentous in nature. Moving on, now we are having one more topic in the fungus, that is Basidiomycetes. It is also called as club fungi. The Ascomycetes we have studied also. Now we are studying Basidiomycetes. The Basidiomycetes means they are also called as club fungi. Their habitat, they are found in soil, logs, tree stumps. Some are parasites, rust and smuts. The mycelium is septate and branched. Asexual spores are generally absent. Vegetative reproduction by fragmentation. Sex organs are absent. Plasmogamy occurs by fusion of two vegetative or somatic cells of different strains, that is dikaryotic mycelium N plus N, and karyogamy and meiosis occur in the basidium. Club shaped, producing four basidiospores exogenous. Basidia are arranged in fruiting bodies called basidiocarps. Example are agaricus mushroom, Astilago smut, and Puccinia rust. Life cycle in the Basidiomycetes, the plasmogamy is the fusion of compatible somatic cells, then that turns into a dikaryotic stage N plus N, then the karyogamy in Basidium, and the meiosis also happens in Basidium, and that's how basidiospores are formed. They are exogenous in nature. Nucleus from strain A and the nucleus from strain B has been coded here to understand that how two different nuclei take part in the life cycle of the Basidiomycetes. Basidium means club shaped. It has parts. One upper part is basidiospore. Four spores are present. Sterigma are the stalks of the basidiospores. Basidium is a club shaped structure, and below that one you will find dikaryotic mycelium. Basidiocarp is a fruiting body. Examples are Ustilago, smut, and Puccinia rust. Stop the video here and study the previous year question. After studying the previous year question, you will understand how the questions have been framed from this topic of the Basidiomycetes, and so that the future expected questions can be understood. The key points you have to remember are Basidiomycetes have septate mycelium, lack asexual spores, and form basidiospores exogenously in Basidia. Important fungi include agaricus, also called as mushroom, astilago, also called as smut, and Puccinia, also called as rust. Moving on, we have Deuteromycetes. Deuteromycetes is also called as imperfect fungi. Only asexual or vegetative states known. Sexual stage either absent or not discovered. When sexual state was discovered, they were moved to Ascomycetes or Basidiomycetes. Reproduced by Conidia asexual. Mycelium septate branched. Many are decomposers of litter, help in mineral cycling. Examples are Alternaria, Trichum, and Trichoderma. You can see the asexual reproduction. The hyphae are septate, mycelium is septate and branched. There is a conidiophore flower-like structure in which there are asexual spores inside the conidia, and that is going into asexual reproduction. The examples are Alternaria. Alternaria common on decaying plant material, plant pathogens. Coletotrichum causes anthracnose disease in many plants, and Trichoderma is a soil fungus. It's a decomposer and is a biocontrol agent. This is very important. Direct question can be asked. You can see here in the previous question what type of questions have been asked from Deuteromycetes. The key takeaways from the Deuteromycetes are: Deuteromycetes are called imperfect fungi because their sexual stage is unknown. They reproduced by conidia. Conidia are asexual spores. Mycelium is septate and branched here, ecologically important as decomposers and contributors to mineral cycling. When sexual stage discovered, reclassified as Ascomycetes or Basidiomycetes. Examples are Alternaria, Coletotrichum, and Trichoderma. Now comes the other kingdom, that is kingdom Plantae. So the kingdom Plantae, it means plants. We already know all these things. One is they are eukaryotic, they are chlorophyll containing autotrophic. Most are autotrophic and they have cellulose made up of they have cell wall made up of cellulose. They include algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. And life cycle shows alternation of generations like gametophyte and sporophyte. The kingdom Animalia, when compared with the kingdom Plantae, heterotrophic, they are eukaryotic, multicellular, and they have no cell wall, holozoic nutrition, and internal digestion is present in this kingdom Animalia, and they have locomotion and they have definite growth pattern also. When it comes to viruses, viroids, and prions and lichens, they are non-cellular, inert crystalline structure outside the living cells. Obligate intracellular parasites use host machinery to replicate. Discovered by Dmitry Ivanovsky. Mosaic disease of tobacco, MW Beijerinnicum, named virus, and Contagium vivum fluidum, infectious living fluid. W.M. Stanley crystallized the TMV, showed viruses are nucleoproteins. Structure is protein coat called capsid made up of capsomeres, and inside either there is an RNA or DNA, never both. Plant viruses usually are single-stranded RNA, whereas the animal viruses you can find both the single-stranded RNA and a double-stranded RNA and also the DNA. The bacteriophages are usually double-stranded DNA, but not always. Viruses cause diseases like mumps and smallpox and herpes and influenza and AIDS, which is caused by HIV and COVID-19. In plant, they cause mosaic formation, leaf rolling, and dwarfing. So after studying the differences between kingdom Plantae, Animalia, Viroids, and Prions and Lichens, you have to see the diagram here. The main thing when compared these kingdom are about the reproduction. Okay, in plant there is an alternation of generations, whereas you can see the structure of a virus here and tobacco mosaic virus, and direct questions can be coming from the image basis also. The key points are viroids. Viroids are free RNA molecules without the protein coat and cause diseases in plants like potato spindle tuber viroid. Prions are infectious proteins basically, and abnormally folded proteins are called as prions and cause neurodegenerative diseases called as, example, mad cow disease. So these examples with their features of these virions and prions are directly exam oriented. You can observe here the previous year questions, how the questions have been formulated in this topic, so that if you integrate these questions with the slide, you'll come to know what are the expected future questions. Viroids. Viroids discovered by T.O. Diner, potato spindle tuber disease, free RNA, low molecular weight and circular, no protein coat, smaller than viruses. So the structure of the viroid is circular, single-stranded RNA, no protein coat. And the key points are they infect plants. Example, potato spindle tuber disease, small infectious non-encapsulated RNA molecules and replicates inside the host plant cells using host machinery. So the structure of the viroid is very important. It is circular. It is single-stranded RNA. There is no protein coat. The key points obviously are that it infects the plants. Example: potato spindle tuber disease. Small, infectious, non-encapsulated RNA molecules replicates inside the host plant cells using host machinery. Prions. When it comes to prions, it's actually abnormally folded prion. There is a conformational. Conformation means shape. The diseases caused by the prions are bovine spongiform encephalopathy (BSE), mad cow disease in cattle, and Creutzfeldt-Jakob disease (CJD) in humans. So the takeaway here are basically virions are infectious RNA, no protein coat, and whereas the prions are infectious proteins, no nucleic acid. Next coming the lichens. Lichens are basically symbiotic relationship association between fungus microbiont plus alga, that is a uh, phycobiont. Alga autotrophic and fungus is heterotrophic. The mutual benefit in the lichens is fungus provides shelter, water, minerals, and alga provides food. The pollution indicators, good indicators of air pollution, do not grow in polluted areas, especially sensitive to sulfur dioxide. Types based on the algal partner. Green algal lichens, most common example Parmelia, and cyanobacterial lichens, less common example Nostoc. Structure of a lichen you have to understand here. The upper layer is called as cortex. After that one there is a fungal hyphae microbiont. Then comes the algal cell, that is phycobiont, and below that one there is medulla, that is loose hyphae and lower cortex. Then after that one you will see some rhizines which are anchor like hyphae. So key points here are lichens are a perfect example of symbiosis. Fungus provides protection and absorption. Alga prepares food by photosynthesis. They are excellent bioindicators of air pollution, especially sulfur dioxide sensitive. So the previous year questions here are being given. Stop the video and study these. Corner concepts, small but testers. In every video, after telling you all the important topics, at last I am going to give you small topics which are usually considered to be less important, but questions are usually seen from that topic. Okay, what is the full form of PPLO? That's an actual second name of mycoplasma. What is the first restriction endonuclease discovered? It was Hind 2. What is the genetic material in bacteriophage T2 DNA, and which bacteria are used in curd formation, that is lactobacillus heterotrophic bacteria? All these 20 topics are corner concepts. They are small facts but are often asked directly in exams, so revise them regularly. Now comes the predicted questions for future NEET. So I have given some of the questions like assertion, reason, match the following in statement based questions and diagram based questions and classification sequence. These questions are expected future NEET questions, not directly or not exactly, but something of this sort are expected. So stop the video here and study all these predicted questions in detail. Now comes the plant kingdom. So from plant kingdom, the highest weightage is from algae, especially three classes of algae: the Bryophytes, the Pteridophytes, and Gymnosperms. And moderately important are life cycle patterns. So out of all the like 2 to 3 questions from plant kingdom, most commonly the questions are coming from algae and Gymnosperms. But next is moderate questions from life cycle patterns. Obviously, this is examiner's blueprint. Obviously, there could be a possibility there more questions could come from a lesser important topic also. I'm just giving you an idea so that while you are preparing, you also prepare with that level of importance. Let's see. Systems of classification: artificial, natural, and phylogenetic. The name itself tells you what and why are these names given to which type of classifications. Artificial systems, it means based on few gross superficial morphological characters like habit, color, number, and shape of leaves and rhizoom structure. This was given by Linnaeus. Gave equal weightage to vegetative and sexual characters. Vegetative characters are more easily affected by environment. Separated closely related species. So this was given by Carolus Linnaeus, and he was the father of taxonomy. This is artificial system. Next comes the natural system. Based on natural affinities among organisms, considered external and internal features, ultrastructure, anatomy, embryology, and phytochemistry, attempts to group organisms reflecting the true relationships. Example, classification of flowering plants by George Bentham and Joseph Dalton Hooker. So he actually used some natural affinities, natural close relationship between the organisms, and considering the external and internal features. Now comes the phylogenetic system. In the phylogenetic system, it is based on evolutionary relationship. So according to the evolution, which organism is related to the another organism, that evolutionary relationship is being explained in the phylogenetic systems. Assume that organisms belonging to the same taxa have a common ancestor. Reflects lineage and evolutionary history. Currently acceptable is phylogenetic system. Next comes the numerical taxonomy, that is also called as phonetic taxonomy. Uses all the observable characteristics. Numbers and codes are assigned to all characters. Data processed by computers to determine the relationships. Each character give equal importance. Next comes the cytotaxonomy. It based on the cytological information. It means the information of a cell. Considers chromosome number, structure, size, shape, arm ratio, and behavior. Next comes the chemotaxonomy. Uses chemical constituents of plants to resolve taxonomic problems. Alkaloids, flavonoids, and terpenoids, etc. So here are some of the previous year questions. So this is about artificial classification, natural classification, phylogenetic classification, and numerical classification, cytotaxonomy, and chemotaxonomy. Let's study about the algae. Almost all parts of the algae have been studied in your previous classes also. Let's study in detail when it comes to NEET importance. So the definition is chlorophyll bearing simple thalloid autotrophic largely aquatic organisms occur in moist stone soils wood in association with fungi as lichens and animals sloth bear. Size ranges from colonial volvox to filamentous Eulothrix and Spirogyra to massive marine forms like kelps. The reproduction, vegetative reproduction by fragmentation, each fragment develops into a thallus. Asexual reproduction by spores, most common are zoospores, flagellated and motile, also aplanospores. The sexual reproduction happens by the fusion of gametes. The sexual reproduction happens by three processes like isogamous, anisogamous, and oogamous. The gametes similar in size are going to meet in this reproduction called as isogamous seen in Eulothrix and Spirogyra. Anisogamous where gametes are dissimilar in size, example Eudorina. And oogamous fusion of a large non-motile female gamete egg and a small motile male gamete sperm, example Volvox and Fucus. The economic importance of these algae, at least half of the total carbon dioxide fixation on earth by algae. So they are primary producers and also helpful in more than half of the carbon dioxide fixation happening on earth. Increase the dissolved oxygen. Food like porphyra, laminaria, and sargassum, 70 species are used as fuel. Hydrocolloids, align from brown algae, algin from brown algae, and carrageen from red algae, agar from gelidium, and gracilaria used to grow microbes in ice creams and jellies. Chlorella unicellular, rich in protein, used as a food supplement in for space travelers. So this chlorella is an algae and it is unicellular, and for the space travelers it is used as a rich protein supplement. Examples across the diversity you have to remember. Colonial example is Volvox. Filamentous algae example is Eulothrix and Filamentous Spirogyra. Massive marine are kelps. Edible red alga is Porphyra, edible brown alga is Laminaria, and edible brown alga is Sargassum. Common terms you should remember without getting confused is zoospores are motile and flagellated spores, aplanospores are non-motile spores, thallus is a body of algae, and hydrocolloids are water-soluble polysaccharides. The key takeaways from the algae are simple, mainly aquatic autotrophic organisms with great ecological significance and economic significance. Understand the reproduction types like isogamy and oogamy, and uses of algae based products like algin, carrageen, agar and chlorella. Stop the video here and study the previous year questions. Classification of algae classified into three classes. Let's see one by one. The first class is Chlorophyceae, also called as green algae. The pigments present in the green algae Chlorophyceae are chlorophyll A and B, called as grass green. Stored food, starch in pyrenoids, proteinaceous bodies surrounded by starch. So basically it is stored as starch, and the shape of storage is as pyrenoids, that is they are proteinaceous bodies surrounded by starch. The cell wall is made up of cellulose as inner layer plus pectose outer layer. Flagella are 2 to 8 equal and apical. It means at the apex part. Top part. Habitat freshwater most, and some are brackish and saltwater also. Examples are Chlamydomonas, unicellular, Volvox, colonial, Eulothrix filamentous, Spirogyra filamentous, spiral chloroplast, and Chara, complex oogamous with globule and nuclei. Reproduction: vegetative by fragmentation, asexual by zoospores, sexual isogamous, anisogamous, and oogamous. Examples of the chlorophyceae are Chlamydomonas. Remember the diagram also. Diagram based questions can come in this topic. Habitat: freshwater most, brackish and saltwater also can be seen. Let's see the examples. Okay, so Chlamydomonas, you can see the diagram here. It is unicellular. It has two flagella are on the top, therefore the flagella are apical. There is one eye spot and there is a cup shaped chloroplast with the pyrenoid. The size of the Chlamydomonas is 10 micrometers. Now comes the Volvox are colonial. They have a lot of daughter colonies, and the size of the colony is 500 micrometers. Eulothrix is basically filamentous. It is 20 micrometer in its thickness, and you can find a part of the Eulothrix is zoosporangium. Spirogyra is also filament. They have spiral chloroplasts and pyrenoids. Chara is complex and oogamous. It has an antheridium, it has an oogonium, it has a globule, nucleole, and internode, and also root-like structures called as rhizoids. Direct diagram-based questions could also be asked. Reproduction in Chlorophyceae mostly vegetative, that is by fragmentation. Asexual reproduction happens by zoospores, and sexual reproduction by isogamous or anisogamous or oogamous also. Key points you have to remember about the Chlorophyceae are: starch is stored in the chloroplast pyrenoids. Chloroplast is cup-shaped in Chlamydomonas. Eulothrix and Chara form zoospores and gametes respectively, whereas conjugation in Spirogyra produces zygospores. So specific to that example also, these are the key points you have to remember for exam. Stop the video here and study the previous year questions, and also after studying the previous year questions, integrate those questions with the content here. Now let's study about the other category of the algae, that is Phaeophyceae, brown algae. Let's see the main differentiating points. When it comes to pigments, it has chlorophyll A and C. Also it has carotenoids and fucoxanthin. Xanthophyll gives brown color. The stored food in the Phaeophyceae are mannitol and laminarin, complex carbohydrates. Cell wall is made up of cellulose plus algin, that is gelatinous coating. There are two flagellae and both are unequal and lateral. Habitat is mostly marine and freshwater, that is rare. Plant body attached by the holdfast has stipe, that is stalk, and frond, that is leaf-like structure. Some kelps reach 100-meter long also. Reproduction: vegetative, asexual, and sexual. In vegetative, obviously the reproduction happens by fragmentation, and in the asexual, by biflagellate zoospores. Pear-shaped unequal flagella are also present in the zoospores. And also in the Similarly, the sexual reproduction is isogamous, anisogamous, and obviously oogamous. Any one of these kind can happen in comes when it comes to sexual reproduction. The examples are ectocarpus, simple branched and filamentous, and dictyota, laminaria, sargassum, and fucus. You can see the diagram here. Diagrams are present in the NCERTs also. The leaf-like structure is called as frond, and the hold fast attachment for attachment, and there is a stalk called as stipe. The asexual reproduction happens through zoospores, and these zoospores are pear-shaped, biflagellate, and they are unequal flagella. And sexual reproduction happens like isogamous, anisogamous, and oogamous. Examples of the Phaeophyce brown algae are Ectocarpus, simple branched filamentous, and Dictyota is you can see here Dictyota, okay, and Laminaria, giant kelp, sargassum, air bladders present in the sargassum, and fucus, these bladders are present. Key takeaways are brown algae are mainly marine, have mannitol and laminarin as stored food, origin in cell wall, and asexual reproduction by biflagellate zoospores. And examples are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. You can stop the video here and study the previous year question. All these previous year questions have been asked previously, and after studying this one, you'll come to know what are the current questions he is going to ask again in the next examination. Moving on, the third category of the algae we are going to study is Rhodophyceae, also called as red algae. Let's see the important key features of this Rhodophyceae, that is red algae. Pigments are chlorophyll A, D, and phycoerythrin, a red pigment, because of which the whole algae is red in color, and phycocyanin. So the pigments basing on the coloring also you have to remember. Okay, stored food here is Floridian starch, that is similar to amylopectin and glycogen, that is called as Floridian starch. The cell wall is cellulose, pectin, and polysulfate esters like agar and carrageen. Flagella is absent. It is non-motile spores and gametes. So habitat is mostly marine. Warmer areas can grow at great depths because phycoerythrin can absorb blue light that penetrates deep also. So the reason why this red algae can be found in the very deep areas also, the reason has been given here. The reproduction vegetative by fragmentation, asexual by non-motile spores, sexual oogamous with complex post-fertilization development. So vegetative by fragmentation, obviously asexual by non-motile because there is no flagella here, and sexual reproduction with complex post-fertilization development. So then comes the sexual oogamous with complex post-fertilization development. This post-fertilization development was never a part of the previous classes. So that is very important. Examples are Polysiphonia, Porphyra, and Gracilaria and Gelidium. Let's see one by one the structural importance here. Polysiphonia has many branches. Poly means many, siphon means a branch. So therefore branching filamentous structure is present in the Polysiphonia. Now comes the Porphyra. It is a sheet-like folios, like it means folio means leaf-like, sheet-like structure. Gracilaria, it has cylindrical branches, thin stick-like cylindrical branches are present in Gracilaria, and for the Gelidium, it has stout cartilaginous branches. It is very important here. Comparison between the tables of the algal classes, you can see that. Okay, in the chlorophytes there is green algae, the uh, Phaeophyce, brown algae, and Rhodophyce algae, red algae. Okay, so the pigment in the chlorophyce is chlorophyll AB, and Phaeophyce is chlorophyll A, C, and fucoxanthin, and the Rhodophyce is chlorophyll A, D, and phycoerythrin. Stored food in the chlorophyce is starch, whereas the stored food in the Phaeophyce is mannitol and laminarin, and the stored food in the Rhodophyce is Floridian starch. And the cell wall is made up of cellulose in chlorophyce, cell wall is made up of cellulose and algin in Phaeophyce, and cell wall is absent in Rhodophyce. Similarly, when it comes to flagella, 2 to 8 equal and apical flagella can be seen in chlorophyce, whereas only 2 flagella, unequal and lateral condition situation is seen in the Phaeophyce, and totally the flagella is absent in Rhodophyce. And habitat, when it comes to habitat, chlorophyce is can be seen in freshwater, brackish, and saltwater also. Whereas in Phaeophyce, freshwater is rare, but brackish and saltwater marine can be seen. And in Rhodophyce, some can be found in freshwater, brackish water, and but mostly they are found in salt water, that is marine waters. Now moving on, the next chapter, the next topic is bryophytes. Okay, so these are the evolved plant. They are also called as amphibians of the plant kingdom, can live in soil but dependent on water for sexual reproduction. Amphibians, it means they can live both in the water conditions also and in the land conditions also. The habitat is they need to have damp, it means they should be wet, humid, there should be some water content, shaded, there should be some shade, direct sunlight exposure is not favorable for them, and important in plant succession on bare rocks. Plant body more differentiated than algae. Thallus like prostrate or erect, attached by rhizoids, unicellular or multicellular. Lack true roots, stem, leaves have root-like and leaf-like, stem-like structures in the plant body. Gametophyte. Gametophyte is a dominant haploid independent uh, structure. It bears sex organs. Those are antheridium, a male structure, produces biflagellate antherozoids, and they have also like archegonium, that is they are female structure, that is flask shaped, produces a single egg, that is called as gametophyte. So fertilization and life cycle needs water for antherozoids to swim to archegonium, that is female structure. Zygote forms the sporophyte. Sporophyte is multicellular, attached to and dependent on gametophyte. Saprophyte has foot, seta, capsule. Spores produced by meiosis in capsule. Spores germinate to form new gametophyte. So economic importance is sphagnum, that is peat moss, used as fuel, peat, and packing material for living plants. Water holding capacity is high. Mosses also prevent soil erosion. There are two groups. The group one is liverworts and the other is mosses. You can see the structure here also. The structure shows specific antheridium, that is male structure, and archegonium, a female structure. Now, moving on, you have to understand about the structure here. Be direct structure or diagram based questions can be asked. Okay, so the antherozoids are biflagellate. You have to see the diagram here. The life cycle of the bryocytes you can see here. There is a new gametophyte which is haploid, which gives rise to antherozoids which have two flagella and they can swim in water. Then they go and fertilized with the an archegonium and zygote is formed, that is 2n diploid, and this it turns into sporophyte dependent on the gametophyte. So capsule sporangium, it has seta and foot also, and that gives rise to spores, and after spores it goes into meiosis and gives rise to new gametophyte, that is haploid. So this sequence and flow of the life cycle also you have to understand. Moving on, the examples are Marchantia, that is liverwort, and Riccia, that is also liverwort. Funaria is a moss, and Sphagnum is also a moss, also called as peat moss. The key takeaways are bryophytes are the amphibians of the plant kingdom with dominant gametophyte and dependent sporophyte. Sexual reproduction requires water, important in succession, soil conservation, and as peat moss, that is sphagnum. Liverworts example is Marchantia. Example. Let's see one by one. General features. Thalloid. The liverworts are thalloid, that is dorsiventral, dorsal side green with air pores and scales, ventral side purple with rhizoids, closely appressed to moist substratum. So wherever there is moist condition, that type of a land surface, they are tightly attached. That is called as closely appressed. Asexual reproduction through gamma. What do you mean by gamma? Green multicellular asexual buds produced in gamma cups on the dorsal surface of the thalli are called as gamma. So gamma are green, gamma are multicellular also. Okay, gamma detach, fall on moist soil and germinate to form the new thalli. So gamma cap produces gamma, and gamma germinating gamma then young thallus. Sexual reproduction in which male and female sex organs may occur on the same thallus, monoecious, or on different thalli, that is dioecious. Sporophyte. After fertilization, zygote develops into sporophyte. Sporophyte differentiated into foot, seta, and capsule. Meiosis occurs in capsule producing spores. So this is about the sporophyte. You can see the diagram here. The thallus of the Marchantia liverfert has air spores. It has scales also. It has gamma cups on the dorsal side, and it has rhizoids on the ventral side. The sex organs of the Marchantia are antheridiophore, that is male thallus, which has antheridia in chambers, and archegoniophore, that is female thallus, which has archegonia in chambers. The sporophyte, after fertilization, the parts are: it has a capsule, spores produced after meiosis, it has a seta, seta is a stalk, and it has a foot embedded in the female thallus. So longitudinal section of the capsule can be seen here, that it has pores and elaters. What do you mean by elaters? Elaters are the structures which aid in the spore dispersal. Elaters are hygroscopic, spirally thickened cells that uncoil in dry air and help in spore formation. Let's see the key points of Marchantia. Marchantia shows dichotomous branching. Gemme cups and antheridiophore and archegoniophore are unique to Marchantia only. Sporophyte is dependent on the gametophyte. Elaters help in the spore dispersal. You can see the previous year questions here also. Stop the video and study the previous year question here itself. Moving on to mosses. Example: Funaria, sphagnum and polytrichum. It has two stages. One is gametophyte and the sporophyte, but the gametophyte itself has two stages now, that is protonema and leafy stage. The creeping green branched filamentous develops directly from spore is called as protonema, and leafy stage develops from protonema as lateral bud, upright slender axis with spirally arranged leaves attached by multicellular branched rhizoids. The sex organs are antheridia and archegonia at apex of the leafy shoots. Fertilization occurs and develops into sporophyte. Zygote develops into sporophyte, which has foot, seta, capsule, more elaborate than the liverworts. Capsule sporangium. Capsule has peristomy teeth for spore dispersal. In the liverworts, we had elaters for dispersal, and here we have the peristome teeth for spore dispersal. Vegetative reproduction by fragmentation and budding on the protonema happens in. So this vegetative reproduction by fragmentation and budding is seen on the protonema in when it comes to mosses. Let's see the structural explanation also. You can see the diagram here. The gametophyte has two stages. One stage is protonema stage and the other stage is called as leafy stage. The protonema stage develops directly from spore, protonema creeping branched and filamentous, whereas the leafy stage has spirally arranged leaves, multicellular branched rhizoids, and it is upright, slender axis from lateral bud on the protonema. The sex organs at the apex of the shoots and theridium are male and archegonium are female. Life cycle of the mosses example has been taken as Funaria. The protonema, which is haploid, gives rise to a leafy gametophyte, which is haploid. Then it has to require water. Then it produces antherozoids, which are biflagellate, and then it gives rise to flagellation, fertilization in the archegonium, and then the zygote is formed, that is diploid, and that turns into a sporophyte, that is diploid, dependent on the gametophyte. Then there is a meiosis in capsule, spore formation. Then there is a dispersal of the spores by peristomy teeth, and the spores which are haploid again develop into protonema. So this is the life cycle. You have to stop the video and study all the parts of the life cycle. The vegetative reproduction happens through fragmentation and the budding. So you can see here there are many previous year questions, that is for your quick revision, and by studying the previous year question you will understand that what type of questions can come in the next NEET examination. The key takeaway are: moss gametophyte has protonema and leafy stages, whereas fertilization requires water. Sporophyte has a foot, seta and capsule. It is attached to and dependent on gametophyte. Capsule has peristomy teeth. Vegetative reproduction occurs on protonema. So importance of mosses. Sphagnum is also called as peat moss, used as fuel, peat and packing material due to water holding capacity. It has been used as a packing material. Mosses prevent soil erosion and help in moisture retention. So this is another use of the mosses, which as you know, because these mosses are attached to substratum, they are tightly attached to the substratum. Therefore these mosses prevent soil erosion and help in the moisture retention. Important in plant succession on bare foods. The two groups of bryophytes you have to remember are liverworts and mosses. You have to study all the previous year questions. By studying the previous year questions, you have to integrate this content of the slide so that you can expect what could be the future questions. Next come the pteridophytes. The more evolved type of plants are pteridophytes. So first we are having thallophytes, that is algae and fungi. Then we are having bryophytes like liverworts and mosses. Now we are having pteridophytes. The general features of the pteridophytes are, firstly, they are terrestrial. In the bryophytes we have studied that they are the amphibians of the plant kingdom. It means they can be seen growing in the water condition also and in the land also. But whereas pteridophytes, they are first terrestrial plants with vascular tissues. Vascular tissues means, vascular means having xylem and phloem like structures, also called as vascular cryptogams. The habitat are, they live in cool, damp and shady places, some in the sandy soil also. Main plant body is a sporophyte, differentiated into true roots, stem, leaves. Leaves may be microphyll as seen in the Selaginella, or macrophylls as seen in the ferns. Vascular tissue, xylem has tracheids but no vessels, whereas phloem has sieve cells but no companion cells. Reproduction in sporophyte. The sporophyte bears sporangia on sporophylls, leaf-like appendages. In some sporophylls form compact strobili or cones, as seen in the Selaginella and Equisetum. Spore mother cells undergo meiosis, gives rise to haploid spores. So I'm just reading here to explain you, but you have to stop the video and read multiple times. Gametophyte, also called as prothallus, it is small, inconspicuous, free-living, photosynthetic, thalloid, beards, antheridia and archegonia, needs water for fertilization. Those are the features of the gametophyte when it comes to pteridophytes. Structure of a typical pteridophyte, example fern, it has leaf-like structures called as frond, and which has fiddle head, that is young leaf, and rhizome, underground stem, and adventitious roots can be seen. The lower surface of the fertile frond has sori. Sori are clusters of sporangia. This is very important. There are two types actually when it comes to homosporous versus the heterosporous. Homosporous, the word itself explains that all spores are similar in size and type. This type of homosporous can be seen in examples like Dryopteris, Pteris, Adiantum, and Equisetum. Whereas heterosporous, there is two types of spores, megaspores large and microspores small. Example, Selaginella, Salvinia, and Marsilia. Megaspore is large and microspore is small. Classes of pteridophytes, as per the NCERT, it has four classes, that is Psilopsida, Lycopsida, Sphenopsida, and Pteropsida. Psilopsida, example is Psilotum. It is small, simple plant. Leaves are absent. Scale-like appendages are present. Sporangia is fused to form synangia. It is called as it is present in Psilopsida, in which the sporangia are fused to form synangia. When it comes to Lycopsidia, examples are Selaginella and Lycopodium. It has microphyll leaves, sporangia on sporophylls, usually in strobili, that is cortex. Cones. Sphenopsida, example Equisetum, horsetail, jointed hollow stem with nodes and internodes, leaves in whorls, scale-like strobili at tips. Pteropsida, example is Dryopteris, Pteris, and Adiantum, that is ferns. Macrophyll leaves fronds, sporangia in sori, usually on the lower surface of the fronds. So these are the classes of pteridophytes you have to remember. You have to remember that strobili at tips and leaves as whorls are seen in Sphenopsida, whereas sporangia in sori is seen in Pteropsida. And this sporangia usually has strobili, but cones are seen in Lycopsida. whorls in Sphenopsida and cones in Lycopsida. These differentiating features are helpful for you to identify the correct answer in the question. You can study the previous year question I have given alongside. The key points here are: first, vascular plants on land. Dominant phase is sporophyte. Vascular tissue, tracheids in xylem, sieve cells in phloem, homosporous and heterosporous forms are present. Heterospory is a step towards a seed habit and includes four classes like Psilopsida, Lycopsida, Sphenopsida, and Pteridopsida. In Psilopsida, the sporangia are fused to form synangia. In Lycopsida, the sporangia are on sporophylls, usually in strobili, cone-shaped, and in Sphenopsida, leaves in whorls and scale-like strobili are at tips. So these are the very important points when it comes to pteridophytes. Let's study about the previous year questions. Okay, in the previous year it has asked all these questions. Stop the video and study. Important points to remember are: Selaginella and Salvinia are heterosporous. Strobili or cones occur in Equisetum and Selaginella. Pteridophytes are vascular cryptogams. Embryo retained on parent sporophyte in heterosporous forms, a step towards seed habit. And prothallus gametophyte, small heart-shaped free-living apogamy, is a prothallus giving rise to sporophyte without fertilization. So we have studied about the classification also. Psilopsida, also called as whisk ferns. The example is Xylotum, and Lycopsida, also called as club mosses. Examples are Selaginella and Lycopsidum, and Sphenopsida are also called as horsetails. The example is Equisetum, and Pteropsida are true ferns. The examples are Pteris adiantum and Dryopteris. Moving on, let's study gymnosperms. The examples of the gymnosperms are Cycas, Pinus, Ginkgo, Sequoia. One of the tallest trees are Sequoia, Cedrus, and Nenetum. The roots are tap roots. In Cycas, coralloid roots specialize associated with nitrogen fixing and cyanobacteria, that is nostoc and anabina, are seen. In Pinus, mycorrhizal association with fungus is seen. The stem is unbranched in Cycas or branched in Pinus and Cedrus. Usually the stem is woody. Leaves are simple or compound in Cycas. Pinnate leaves persist for years. Conifers, that Pinus and Cedrus, needle-like leaves with thick cuticle and sunken stomata. Xerophytic adaptations can be seen. So examples of gymnosperms are Cycas, Pinus, Ginkgo, Sequoia, Cedrus, and Neetum. Reproductive structures are strobili as per cones. The male cone is called as microsporangiate and the female cone is called as megasporangiate. Ovule is naked here. Arrangement of cones. Pinus is monoecious and Cycas is dioecious. Male and female cones on the same tree, that is Pinus, monoecious, and male and the female cones on different plant, that is dioecious in Cycas. The life cycle simplified: microsporangium in the male cone, megasporangium ovule in the female cone. Megaspore mother cell is diploid, which undergoes into meiosis and produces the haploid structures. Pollen grain, the male gametophyte, highly reduced. It has outer wall called as exine, the inner wall is called as intine, and the tube nucleus can be seen in the pollen grain. There is a male gametophyte of these gymnosperms. Pollen tube carries male gametes. Seed is naked seed on ovuliferous scale. It has testa, seed coat, haploid endosperm, gametophytic tissue, and embryo which is diploid. The characteristic features here are: heterospores produce microspores and megaspores. Sporophylls arranged in strobili, that is cones. Pollen grain is a male gametophyte which is highly reduced. Female gametophyte within the megasporangium ovule produces archegonia, two or more. And xylem lacks vessels except in neetum. It is advanced and has tracheids. Phloem lacks companion cells, has sieve cells and albuminous cells. Root modification: Cycas, coralloid roots associated with nitrogen fixing bacteria, is cyanobacteria. that is a root modification. Xerophytic adaptations in the conifers. Needle-like leaves can be seen, thick cuticle, sunken stomata, and resin canals and reduced transpiration are the xerophytic adaptations in the conifers. Previous year questions can be seen here. Important points are endosperm is haploid formed before fertilization, pollen tube is present, no double fertilization, and pollen grains released and carried by wind, that is called as anemophily. The gymnosperms. So this is the revision slide for the gymnosperms. You can see here male cone has been shown in the diagram here. The male cone also called as microstrobilus. There are many microsporangia on the sporophylls producing pollen grains, that is male gametophytes. Female cone called as megastrobilus. Ovules on ovuliferous scales produce megaspores, female gametophyte. The structure of the seed is also important. It has a wing developed from ovuliferous scale. It has a seed coat derived from the integuments. It has pollen chamber cavity where pollen grains are stored after pollination. It has a female gametophyte, that is endosperm haploid embryo, usually with two cotyledons. Important point. Seeds are naked, not enclosed in fruit. Endosperm is haploid. Pollen chamber cavity in ovule that stores pollen grains after pollination. Cycads have motile multiciliate sperms like pteridophytes. Pinus seed requires mycorrhizal association for germination. Resin and turpentine are obtained from the pinus. That is economic importance you can see here. Moving on, let's see the adaptations of the conifers. The adaptations are needle-like leaves with thick cuticle to reduce the transpiration. It has sunken stomata. The transfusion tissue helps in conduction and storage. Mycorrhiza help in absorption of water and minerals. Example, pinus. The transfusion tissue in the leaf is seen here. There is a vascular tissue surrounded by parenchyma. The economic importance are timber is produced from cedrus, pinus, and sequoia. Resin and turpentine from pinus. Fuel and paper pulp. Ornamental plants. Cycas seeds like sago edible after processing. The examples of the gymnosperms are cycas, pinus, cedrus, sequoia, and neetum. Sequoia, also called as redwood. So previous questions are also been given here. Gymnosperms are seed producing vascular plants with naked seeds. Heterosporous reproduction reduced dependent on gametophytes and haploid endosperm. Conifers are well adapted to the harsh conditions. Now comes the angiosperms, that is flowering plants. Most common plants which we see are flowering plants, angiosperms. So this should be easy to understand. Key features are seeds enclosed in fruits, ovules enclosed in ovary. Flowers are the reproductive structures. Size, smallest is Wolfia and the tallest is Eucalyptus, over 100 meters. There are two classes, dicotyledons, that are two cotyledons, and monocotyledons, which has only one cotyledon. Economic importance is food, fodder, fuel, and medicines. Wolfia is the smallest plant, sunflower is dicot, rice is a monocot, mango is fruit seed enclosed, and eucalyptus is the tallest tree. So you have to compare the life cycles of all here. Okay, algae, brown algae, bryophyte, pteridophytes, gymnosperms, and angiosperms. The dominant generation in algae is haploid. The dominant generation in the brown algae is diploid, and the dominant generation in bryophytes is haploid gametophyte, and the dominant generation is in pteridophytes is diploid sporophyte, and the dominant generation in the gymnosperms and angiosperms is also diploid sporophyte. The gametophyte is free living and dominant in algae, reduced and dependent on brown algae, dominant and independent on bryophytes, small independent prothallus in pteridophytes, highly reduced pollen grain embryo sac in gymnosperm and angiosperms. Sporophyte, zygote only one-celled in algae, dominant in brown algae, dependent on the gametophyte in bryophytes, dependent on independent in the dominant and independent in the pteridophytes and dominant in gymnosperms and angiosperms. Life cycle type is haplontic cycle in algae, diplontic cycle in the brown algae, haplodiplontic in the bryophytes, haplodiplontic with dominant sporophyte in the pteridophytes and diplontic in the gymnosperms and angiosperms. The previous year questions has been given here. The key points are alternation of generations is universal in plants. The dominant generation differs among plant groups. Bryophyte dominant gametophyte, pteridophytes dominant sporophyte. Gymnosperms and angiosperms highly reduced gametophytes. Life cycle is diplontic. Water is essential for fertilization in non-seed plants like bryophytes and pteridophytes. Corner concepts, usually we ignore these concepts, but directly questions are coming from here. You have to stop the video and study all these 20 concepts. For example, what is the ploidy of the endosperms in gymnosperms? It is haploid. What is mycorrhiza? What is coralloid root? What is peat? What is the function of gamma? All these corner concepts have been asked as a direct question. Predicted future questions from this chapter, that is plant kingdom. Stop the video here and study all the questions here on the display in the slide. These are the expected future questions for your next NEET exam. You have to be thorough in it. From the animal kingdom, the highest probability questions are coming from the phylum Annelida, Arthropoda, Mollusca, Echinodermata, and Chordata. Other chapters are less important, but obviously the examiner can give of his own wish. So nothing is going to be like fixed here. You can see the key features also, like from Polyphera, what is he asking, what are the examples being asked, from Cyclostomata, what has been asked, and from Aves also what has been asked us. To give you a generalized idea, this slide is for you to understand the whole importance of the chapter. Before we are going to enter inside the chapter, stop the video and study the whole slide in detail. This is the examiner's blueprint. Basis of classification in animals. So this basis of classification in animals is actually very easy. Almost all the biology students knows this one. We just have to go through it superficially and also compare actually. Comparison is very important. So levels of organization, we know that there is a cellular level of organization, tissue level, organ level, and organ system level. The cellular level is seen in the porifera, that is sponges, and the tissue level is seen in the cylindrata, nidaria, and ctenophora. The organ level can be seen in the platyhelminthes and other higher phyla, except the next level. Organ system level can be seen in the annelids, arthropoda, mollusca, echinodermata, hemichordata, and chordata when it comes to vertebrates and invertebrates. The organ system level has special features. Digestive system, incomplete digestive system, single opening mouth and anus, same platyhelminthes. Mouth and is anus have a single opening, as a single opening. The complete digestive system, mouth and anus is separate from ascalminthus onwards. And circulatory system, open the pumped open circulatory system, blood pumped out of heart. Cells and tissues directly bathed in blood can be seen in arthropoda, mollusca, and hemichordata. Closed circulatory system, blood circulates through vessels found in annelida, echinodermata, and chordata. So the phylum level of organization and digestive system. So porifera, cellular, cylindrate, cylindrata, nidaria, tissue level of organization and digestive system through is complete. You have to see all those things. Stop the video and compare all the phyla. Let's start with symmetry. What do you mean by symmetry? Symmetry refers to the arrangement of similar body parts on either side of a dividing plane or around a central axis. So, how are the body parts arranged? And according to the body parts, if their arrangement can divide that body into equal halves, or what type of situation, that type of arrangement is called as symmetry. So we have different type of symmetry. Let's see one by one. First is asymmetrical. It means no plane divides the body into equal halves. Example, sponges, phylum Porifera. You can see that even if a plane, an imaginary plane can be given to that organism, then it cannot be formed into a symmetry. The examples are Sycon, Spongilla and Euplectella. You have to remember these examples because directly symmetry versus example of the organism match the following could be expected question from here. Next is a radial symmetry. The word itself also explains you at some level. Radial symmetry. Any plane passing through central axis divides the body into two identical halves. So the plane is passing through central axis and it is dividing the body into two identical halves. Examples of the radial symmetry are coelenterate, ctenophores and echinoderms, adults especially, suitable for sessile or floating life. You can see that several planes pass through the central axis here in coelenterate, ctenophores. Okay, and even in the echinoderms when they are adult. Okay, like pentaradial in adults, example asterias. The example of ctenophore also pleurobrachia, and the example of cnidarian being hydra. Examples are Hydra aurelia, a jellyfish, Pleurobranchia, sea walnut, and Asteria, starfish, Echinus, sea urchin. Bilateral symmetry. Body can be divided into identical left and right halves only in one plane, that is median plane. Examples are Annelida, Arthropoda, Mollusca, Platyhelminthes, Aschelminthes, etc. So Annelida, Ferretima, Arthropoda, Locusta, Mollusk, Pila, Platyhelminthes, Planaria, and Aschelminthes, Ascaris. These are the examples of bilateral symmetry. Comparison between one and another, let's see what happens here. As in the asymmetry, there is no plane, the body arrangement is irregular, and the example is sponges or porifera, and adaptive advantages is fits in the irregular spaces, maximum surface area, that is an advantage. For the radial symmetry, there are many planes or through central axis, parts arranged around the central axis, coelentrates, ctenophores, and echinoderms. Adults are the examples of radial symmetry, useful for sessile or floating organisms, sense stimuli from all directions. Bilateral symmetry, one there is one plane, that is median plane of symmetry, and body arrangement is left and right sides and they are similar, and the examples are annelids, arthropoda, mollusca, platyhelminthes and aschelminthes. Active movement in a particular direction, specialization of head end is seen as an advantage of the bilateral symmetry. We have to note one thing, that echinoderms show bilateral symmetry in the larval stage and radial symmetry, that is pentaradial symmetry, in adult stage. So echinoderms show two symmetries, bilateral in the larval stage and radial in the adult stage. You can study the previous year questions given in the corner in this slide. Now comes the organization, especially tissue organization, that is diploblastic and triploblastic organization and coelom. Let's study one by one. The diploblastic organization, two germ layers, ectoderm external and endoderm internal. An undifferentiated layer, that is mesoglia, is present in between. Examples of the diploblastic organization are Coelenterata and Ctenophora. You can see in the Coelenterata example Hydra, it is diploblastic, therefore there is an ectoderm and there is an endoderm and there is undifferentiated layer called as mesoglia between those two. In the Ctenophora also you can see this same thing. Three germ layers are present in the triploblastic tree or tri for three germ layers, ectoderm, that is external, mesoderm, that is middle, and endoderm, that is internal, from platyhelminthes to chordatum. So triploblastic example, earthworm cross section, you can see here there is an extra layer in the middle that is called as mesoderm. Examples are platyhelminthes, aschelminthes, annelida, arthropoda, mollusca, echinodermata and chordata. Let's see what do you mean by coelom. Coelom means presence of a body cavity. So there are three categories mainly. There are another categories also, but these are the main categories. One is acoelomate, second is pseudocoelomate, and third is coelomate. So in the acoelomate, the word itself tell you that it has no body cavity. Mesoderm is solid. Example are platyhelminthes or flatworms. Example planaria. The transverse section of the planaria has also been given here. You can see that the mesoderm is solid and therefore there is no cavity formation. In the pseudocoelomate body cavity, a pseudocoelom not lined by mesoderm is present. It means there is a cavity, but that is not cavity lined by mesoderm. That is why it is called as pseudocoelom. Mesoderm present as scattered pouches. Example Ascalminthus. Example Ascaris. The transverse section of Ascaris, that is Ascalminthus, ectoderm, mesodermal pouches, pseudocoelom, and endoderm gut can be seen in the transverse section. But in the coelomate, that is eucoelomate, it means true coelom because it is lined by the mesoderm peritoneum. There is a body cavity. Example Annelida, example earthworms. So the key points we have to remember is diploblastic animals have two germ layers and no true mesoderm. Triploblastic animals have three germ layers and has the ectoderm, endoderm, and mesoderm. Coelom types are acoelomates, which no body cavity, pseudocoelomates, false coelom, and coelomate, where we have true coelom. From platyhelminthes onwards, animals are triploblastic. Annelids, arthropods, mollusks, echinoderms, hemichordates, and chordates are true coelomates. So these are one of the basics topics actually one should be knowing from their 10th or intermediate level itself. So you have to revise all this integrated with the previous year questions. Let's see what do you mean by segmentation called as metamerism. Body divided into a series of segments, metameres with serial repetition of the organs. So body is divided into segments one. That segment is called as metameres, and there in that one each segment there is a repetition of the organs present inside that organism. Such type of segmentation and such type of series of segmentation is called as metameres, found in Annelida, Arthropoda, and Chordata. In chordates, segmentation is visible in vertebrae, ribs, nerves, blood vessels, etc. Let's see one by one. Annelida. Example earthworm. There are segments. Segment 1, segment 2, segment 3, and it reaches up to segment end. And these are similar segments, and there are many repetition organisms. Arthropoda. Example prawn. And arthropoda, you can see there is a head part, there is a segmented thorax part, and there is a segmented abdomen part. So the segmented body with the jointed appendages are the characteristic features of arthropoda. Then comes the chordata. You can see the example of the segmentation or the repetition is seen in the vertebral column. There are many vertebrae which are similar, and there are ribs which along with them, and there are spinal nerves and blood vessels going along with them. So segmentation in vertebra, ribs, nerves, and blood vessels can be seen in chordata. So there are some previous year questions I have given here. You can integrate those questions and study. The key points when it comes to metamerism are: metamerism means repetition of the body segments and organs. It is a sign of advanced body plan and division of labor. In chordate, segmentation seen internally, vertebrae, muscles, nerves, and blood vessels. Examples of the segmentation are being given here. Now let's study the notochord, a mesodermally derived. So what is the derivative layer of the notochord? It is a mesoderm. So a mesodermally derived flexible rod-like structure present on the dorsal side of the body during the embryonic development is called as notochord. So animals with notochord are called as chordates, and animals without notochord are called as non-chordates. Porifera to echinodermata, hemichordata has stromochord, not a true notochord. So notochord in the embryo you can see in the diagrammatic format also. The comparison between the chordates and non-chordates. In the chordates, notochord is present, and in the non-chordates it is absent. In the chordates, the central nervous system is dorsal, hollow, single tubular nerve cord, whereas in non-chordates it is ventral, solid, and double. That is, there is nerve cord with ganglia. Pharynx is perforated by the gill slits at least in some stage in the chordates, and in the non-chordates gill slits slits are absent. Heart is ventral below the gut in case of chordates, and heart is dorsal if present in the non-chordates. Post-anal tail is present at least in the embryonic state in the chordates, and in the non-chordates it is absent. You can study the previous year question and integrate the topics with the topic given here. Take home message here is metamerism segmentation shows serial repetition of the body parts in Annelida, Arthropoda and Chordata. Notochord is the defining feature of the chordates and is the basis of their advanced organization. Notochord present in chordates and absent in non-chordates and vertebral column is actually a modified notochord. Moving on, classification of animals phylum wise. So let's study one by one. Phylum Porifera, also called as cellular sponges. So this cellular Porifera is the primitive phylum, and the level of organization is cellular, asymmetrical, mostly body is asymmetrical symmetry, and there is no body cavity, therefore there is no coelom. They are called as acoelomate, and the digestion is actually intracellular digestion is absent, and circulatory system is also absent, respiratory system is absent, whatever exchange takes place through diffusion. Distinctive features are water canal system with pores called as ostia, central cavity called as spongocoel, and opening called as osculum. Don't confuse the spongocoel with the actual coelom, it is a false appearance. Choanocytes, also called as collar cells, line the spongocoel and canals, generate water current and capture food. Skeleton of the spicules, that is calcareous or siliceous or spongin fibers, are present in the sponges, that is called as Porifera phylum. Reproduction: asexual by fragmentation, sexual, hermaphrodite, internal fertilization, indirect development, larva. Examples are Sycon, Skypha, and Spongilla, freshwater sponge, Euplectella, Venus flower basket, and Euspongia, bat sponge. These examples are very important. Direct name and common name could be asked as match the following. You can see the examples of the sponges here. Sycon or Skypha is a marine sponge, and Spongilla is a freshwater sponge. Euplectella, also called as Venus flower basket, and Euspongia, also called as bath sponge. Now, general structure of a sponge, you can see the structure of a sponge has these parts. The upper opening is osculum, the spongocoel is the body cavity, choanocytes are also called as collared cells, and ostia are the pores present on the body surface. Choanocyte or collar cell, it has a flagellum outside, a collar called as microvilli, there is a nucleus inside, and a food vacuole. Generates the water current and captures the food by the choanocyte. The function of the choanocyte is to generate the water current and also to capture food. Canal system types in Porifera. Ascon type is simplest, Sycon type is more complex, and Leucon type is most complex. Skeleton of the sponges: calcareous spicules, that is made up of calcium carbonate, and siliceous spicules made up of silicon dioxide. Spongin fibers form a flexible skeleton. There are many previous year questions I have given on the slide. You have to observe carefully and study. The key points are: poriferans are cellular level organized animals. They possess a water canal system for circulation of water. Choanocyte, that is collared cells, are characteristic of sponges. Sponges are acoelomate and lack digestive, circulatory and respiratory systems. High regenerative power is a universal characteristic of sponges. Phylum Coelenterata, also called as Cnidaria. Silent features are: the level of organization is tissue level of organization, and the symmetry is radial symmetry. Coelom is absent, they are acoelomate. The digestive system is incomplete. Gastrovascular cavity with single opening mouth is present. Distinctive features like cnidoblast or cnidocytes containing nematocysts, stinging capsules on tentacles and body, used for anchorage, defense, and prey capture. Diploblastic with mesoglea between the ectoderm and the endodome is a characteristic distinctive feature of the Coelenterata, also called as Cnidaria. Polymorphism is also a very characteristic feature. Polymorphism means the body of these cnidarians can occur in two different forms. The two body forms are polyp and medusa. Polyp is a sessile cylindrical stage, and whereas the medusa is umbrella shaped and free swimming. Some exhibit metagenesis, alternation of generations. Polyps polyps produce medusa asexually and medusa produce the polyps sexually. Example, obelia. The two body forms you have to compare directly. Polyp is sessile and medusa is a free swimming. Polyp is cylindrical, medusa is umbrella shaped. Polyp has mouth and tentacles and medusa also has mouth and tentacles but directed downwards. Attached to the substratum is polyp, free swimming is medusa. Examples are hydra and adamsia, and the medusa example is aurelia. Nidoblast, nidocyte and nematocyst. You can see here there is a nidocyte with a nematocyst inside. Discharged nematocyst looks like this one, thread everted. The trigger chemical and mechanical stimulus, stinging thread with barbs used for anchorage, defense and prey capture. The metagenesis in Obelia, that is alternation of generations, can be seen in few organisms of the Cnidaria. The example is Obelia. The other examples are Hydra polyp only freshwater, Aurelia jellyfish, Medusa marine, and Physalia Portuguese man of war, colony of polyps and Medusa marine, and Adamsia, that is sea anemone polyp marine, Penatula sea pen colonial polyp marine, and Gorgonia sea fan colonial polyp marine, Meandrina, that is brain coral colonial polyp marine. So you should understand what is the name of the polyp and what is the common name of these cnidarians and where do they stay. Are they freshwater or marine? You can see the only freshwater is the hydra, all the other are marine. So the key points are diploblastic animals with mesoglia are Coelenterata or Cnidaria. Radial symmetry is suited for sessile or floating life. Nidoblast with the nematocysts are unique to Cnidaria. Polyp is sessile. Medusa is free swimming. Metagenesis in many forms. Example, Obelia. Habitat both marine and freshwater, and the most common freshwater is Hydra. The take-home message here is Coelenterata, that is Cnidaria, are diploblastic, radially symmetrical animals with stinging cells called as nematocysts. They show polymorphism, that is the presence of polyp and the medusa status in life, and metagenesis in many forms, and include both marine and the freshwater species. Now comes the phylum Ctenophora, also called as comb jellies. The level of organization is tissue level of organization, radial symmetry, sometimes biradial symmetry, biradial symmetry, and the coelom is absent, that is they are acoelomate, exclusively marine, diploblastic, present both extracellular in the gastrovascular cavity and intracellular type of digestive system. Distinctive features are 8 external rows of ciliated comb plates for locomotion. So the most important feature of the Ctenophora comb jellies is 8, the number 8 external rows of ciliated comb plates for locomotion, that is for the movement. Bioluminescence is well marked. Lack the nidoblast or stinging cells are not present. Two tentacles with adhesive coloblast are present as a distinctive feature of the Ctenophora. You can see in the diagram there is an apical organ. There are there are 8 comb plate rows. There is a gastrovascular cavity. There is a mouth tentacle and coloblast adhesive cells on the tentacles. Reproduction is only sexual, that is hermaphrodite, external fertilization and indirect development. So examples are pleurobrachia and tenoplana. Comb plates or cilia, 8 longitudinal rows of ciliated plates, steens, beating of cilia helps in locomotion. Bioluminescence, light is produced by specialized cells, that is photocytes present in the body. Two planes of symmetry at right angles to each other. So the life cycle is adult hermaphrodite, gametes, sperm and egg. Fertilization is external and ciliated larva is Cyripid larva and the young is called as ctenophore. The examples are pleurobrachia and ctenoplana. The key points here to remember are ctenophores are exclusively marine diploblastic animals. There is no freshwater ctenophore. They have 8 rows of ciliated combo plates for locomotion. Bioluminescence is well developed. They lack nidoblast, possesses coloblast on tentacles, reproduce only sexually. Fertilization is external. Phylum Platyhelminthes or flatworms. So in the flat forms, that is platyhelminthes, the level of organization is organ and sometimes organ system also, bilateral symmetry, and they are acoelomate. It means there are few characters which are advanced and few are primitive in nature. The digestive system is incomplete. There is a blind sac plan, except some have no digestive system, that is parasitic forms absorb nutrients through the body surface. Digestive distinctive features of the platforms are they are dorso-ventrally flattened body, they are triploblastic. Parasitic forms have hooks and suckers, flame cells, that is solenocytes for osmoregulation and excretion, high regeneration capacity in the planaria. So we have studied the regeneration capacity in the porifera also, and here in the flatworms example planaria there is a high regeneration capacity. Reproduction is a hermaphrodite, that is monoecious, internal fertilization and many larval stages. Examples are Taenia, tapeworm, fasciola, liver fluke and planaria free living. You can see the body here being dorsoventrally flattened body plan. There is an eye spot on the top. There is an auricle. Pharynx is aversible and gastrovascular cavity, that is incomplete blind sac can be seen there, and the mouth is on the ventral side. Flame cell or solenocyte. It has a ciliated flame bulb inside which there is a tubule cell, nucleus and excretory pore. Flame cells help in the osmoregulation and excretion. The important examples are Taenia. There is a tapeworm. Tapeworm has a scolex hold. The scolex is helpful to hold to intestine. There is a neck and there are many number of proglottides. Endoparasite in the human intestine, Taenia or Taenia saginata or Taenia. There are many types of Taenia actually. They are endoparasites in human intestine. Body is segmented into proglottides. Each mature proglottid is hermaphrodite. No digestive system. Absorbs nutrients through tegument present on the surface. Two species, Taenia solium with hooks and Taenia saginata without hooks. Taenia solium is a pork tapeworm and Taenia saginata is a beef tapeworm. Fasciola, also called as liver fluke, has one oral sucker and another ventral sucker. There are branching intestine, blind sac is seen, endoparasite in the bile duct of the sheep, cattle and humans. Single opening, that is mouth is equal to anus, hermaphrodite. Intermediate host is limnia, that is snail, and larval stages are miracidium, sarcaria and metacercaria. You should remember the names of the larval stages here. When it comes to planaria, it is not a parasite, it is a free living. In freshwater, high power of regeneration, only a small piece can regenerate into a new individual. Useful model for studying the regeneration. You can see the eye spots, auricle, aversible pharynx, and the gastrovascular cavity in planaria. The life cycle of the Fasciola hepatica digenetic is eggs in the feces of the infected host turns into miracidium and hatches into sporocyst in snail and redia in snail and cercaria in snail and metacercaria on vegetation in water. Adult in the bile duct of the host sheep, cattle or humans. In the intermediate host snail limnia, and in the primary host is definitive host. There are many previous year questions you have to study carefully. Parasitic adaptations of the platyhelminthes. Mainly the presence of hooks and suckers for attachment. Dorso-ventrally flattened body is also a parasitic adaptation of the platyhelminthes. Tegument is resistant to dignitive host, to digestive enzymes of the host, and it has highly developed reproductive system and many eggs, reduced or absent digestive system, flame cells for osmoregulation and excretion. Regeneration in planaria happens cut into pieces. Each piece regenerates into a complete new individual. The key points of the platforms are triploblastic, bilaterally symmetrical, acoelomate animals, incomplete digestive system, and flame cell solenocytes are characteristics excretory organs, hermaphrodite with internal fertilization, important economically as parasites causing diseases, example teniasis and fasciolasis. So the phylum Ascomynthus nematoda or roundworms. Let's see direct comparison also. because we have already studied the previous four phyla, then obviously you should know that at which points we are going to cover. So organ system level of organization: bilateral symmetry, coelom is pseudocoelomate, free, free living in moist soil, water, or parasitic in plants and animals, elongated, cylindrical, and unsegmented tapering at both ends, complete alimentary canal with muscular pharynx, and circulatory system is absent. Respiratory system is absent, gaseous exchange by diffusion. Excretory system: excretory tube, Renate cell or H cell with excretory pore. Sexes separate, that is dioecious, females usually longer than males. Reproduction, fertilization is internal, development is direct or indirect. Nervous system: nerve ring around the pharynx with longitudinal nerve cords. Distinctive features are circular cross section, triploblastic, thick non-cellular cuticle. Pseudocoelom acts as a hydrostatic skeleton. Examples are Ascaris roundworm, Wuchereria bancrafti, filarial worm causes elephantiasis, Ancylostoma vermicularis, hookworm, and Enterobius pinworm. You can see the diagram also. The cross section is also been given here. You have to stop the video and study this diagram in detail. There is excretory pore, excretory canal, and Renate cell, H cell in the excretory system. Reproductive system is also has given here. Male is smaller, female is larger. There are spicules and testes. There are vas deferens and cloaca. Examples important to human parasites are Ascaris lumbricoides, roundworm causes ascariasis, Wuchereria bancrofti, filaria worm causes elephantiasis, and Ancylostoma duodenale hookworm causes anemia, and Enterobius vermicularis pinworm causes enterobiasis. Key points are Ascalminthus are triploblastic, bilaterally symmetrical animals. They have pseudocoelom, not a true coelom. Digestive system is complete with muscular pharynx, excretion by H cell, Renate cell, and excretory pore. And sexes are separate. Fertilization is internal, and development is direct or indirect. Phylum Annelida, segmented worms. Organ system level of organization is seen in the phylum Annelida. Bilateral symmetry is seen. True coelom or schizocilous coelom is seen. Metameric segmentation, that is body divided into segments, metamerism is seen. Complete digestive system, closed circulatory system, nephridia are part of the excretory system. Paired ganglia, double ventral nerve cord, nervous system. Nereis is dioecious, earthworms and leeches are monoecious, that is hermaphrodite. Longitudinal and circular muscles and aquatic forms have parapodia nereis as part of the locomotory structures. Examples are nereis marine, Ferretima earthworm, and Hydrodinaria leech. You can see the internal organization in the cross section of the earthworm. Ferretima, the outermost layer is cuticle followed by epidermis, circular muscle, longitudinal muscle. There is a dorsal vessel, intestine, nephrostome, septal nephridium, coelomic cavity, ventral vessel, and nerve cord. Segmented body with true coelom, schizocilous. Each segment metamer has a complete set of organs. The anatomy of the earthworm has been also shown in the picture. Prostomium is first, peristomium, then comes the scutellum, and then the anus. Nearly 30 segments can be seen there. Digestive system is complete with mouth, esophagus, and esopharynx, gizzard, intestine. Circulatory system is closed with dorsal vessel, main heart, and ventral vessel, lateral heart, and excretory system has nephridia. Nephrostome is the opening, coelomic cavity, nephric tubule, and nephridiopore. Excretory pore can be seen. Examples of the annelids are Nereis. Nereis is dioecious, it is marine, it is free living, it has parapodia with setae for locomotion and respiration. Ferretima earthworm has mouth and anus and clitellum. Hermaphrodite, it is monoecious. Terrestrial, lives in soil, important in soil aeration and humus formation. Hydrodinaria, leech, hermaphrodite, monoecious, aquatic, ectoparasitic, anterior and posterior suckers for attachment. So, types of nephridia in the earthworm: pharyngeal nephridia, septal nephridia, and integumentary. So, The pharyngeal nephridia are seen in the first few segments, and septal nephridia are seen in the most segments, and integumentary nephridia are seen in the last segments. Locomotion is by muscles in earthworm, and in the needles there are parapodia. So there are many previous year questions also given here. You have to analyze these questions and also see the diagram, cross section, segments multiple times in order to answer the questions properly. Phylum Arthropoda, the largest phylum. It has organ system level of organization. The symmetry is bilateral symmetry. Coelom is true coelom. Open circulatory system, hemocoel. Segmentation, segmented body with jointed appendages can be seen. Exoskeleton, chitinous with proteins. Body divisions, head, thorax and abdomen. Cephalothorax in some. Respiration, gills, book gills and book lungs. Tracheal system, excretion, Malpighian tubules, insects, green glands, crustaceans. And nervous system, ventral nerve cord, reproduction, dioecious, internal fertilization and mostly oviparous. Body plan generalized in arthropods. The head, thorax and abdomen are having segments, and there is appendages also. Three pairs of walking legs on thorax, two pairs of wings on thorax, and segmented abdomen with appendages can be seen. Respiratory organs are in prawns there are gills, in linulus there are book gills, and in the scorpion there are book lungs, and in the grasshopper there is a tracheal system. Excretory organs in the cockroach is Malpighian tubules, and in case of prawns they have green glands. Circulatory and nervous system, it has open circulatory system, a dorsal heart with ostia and a hemocoel open body cavity and blood sinuses. Ventral nerve cord is seen in cockroach, brain, supraesophageal ganglion and subesophageal ganglion and ventral nerve cord with ganglia. Important examples here are Insecta, Apis honeybee, Bombyx silkworm, Locusta locust and cockroach. Arachnida scorpion and spider, Crustacea paleomon, Limulus prawn and king crab, Myriapoda that is centipede and millipedes. Others are Lacifer, lac insect and Anopheles mosquito. Arthropod means jointed appendages, largest phylum with maximum species of diversity. Exoskeleton made of chitin protects and prevents the water loss. Open circulatory system with dorsal heart. Excretion by Malpighian tubules. Insects, green glands are present in the crustaceans. Similarly, phylum Mollusca, it is second largest phylum after the phylum Arthropoda. As we have seen in the earlier slide, phylum Arthropoda, which has jointed appendages, is the largest phylum when it comes to diversity of species. The level of organization here is organ system of level of organization. The symmetry is bilateral. The coelom here is a true coelom, that is reduced one, and the digestive system is complete. It means it has both opening and also an exit opening. Mouth and anus. Circulatory system is open type. Most mollusks have open type. Few we are having exceptions. Their distinctive features are soft body usually covered by calcareous shell. Calcareous, it means it is made up of calcium carbonate. Body is unsegmented with distinct head, muscular foot and visceral hump. Mantle covers visceral hump. Mantle cavity contains feather-like gills. Radula like file-like rasping organ in mouth absent in the bivalves. Respiration is by gills, and some have ctenidia. Excretion is by nephridia. Reproduction usually dioecious. They are oviparous. Indirect development, trochophore or veliger larva is seen during the development. That is why they are called as indirect development. Example: Pila pinctada, Sepia, Loligo, Octopus, Aplysia, Dentalium, and Chetoplura. Now comes the phylum Echinodermata. They are also called as spiny skinned animals. Echino means spiny and derma means skin. So these animals have a special feature that they are spiny skinned. Now let's go into the general characters. The level of organization is organ system. Symmetry in adults is radial, that is pentamerous radial symmetry, and in the larvae it is bilateral symmetry. The coelom is true coelom, enterocoelous habitat, gut is exclusively marine. The body wall is endoskeleton of calcareous ossicles or spines in the dermis. Digestive system complete, mouth on the ventral side and anus on the dorsal side. Circulatory system present but reduced, poorly developed respiratory system through dermal branchiae, papillae and tube feet. You have to remember the respiratory system. It is very important. Excretory system is absent. Nervous system is nerve ring and radial nerves. Reproduction is dioecious, external fertilization, indirect development, free swimming larva. The distinctive features are water vascular system, unique to echinoderms, helps in locomotion, capture and transport of food, respiration, tube feet, podia with suckers, spines present on the body surface. You have to see the diagram. The diagram shows that there is madreporide, that is a sieve plate, then there is a stone canal, there is a ring canal, there is a radial canal, and there is a lateral canal. The tube feed podia with suckers can also be seen here. Madreporide leads to stone canal, leading to ring canal, then leading to both radial canals and ending into the tube podia. And in the tube foot, that is podia, there is ampulla, and then at the last there is suckers. So this functions for the locomotion, capture and transport of food and respiration. This is called as water vascular system. It is unique to echinoderms. This water vascular system, you have seen this water canal system in sponges, porifera, but here it is having water vascular system. It is unique to echinoderms. The functions are locomotion, capture and transport of food and respiration. The structure of a typical starfish asterias is it is having anus on the dorsal side, it is having spine for each arm of the starfish, the digestive glands are present, there is a pyloric cecum, there is a mouth on the ventral side, and the tube feet, podia can also be seen in the picture. This picture and memorization of this picture is very important. The life cycle of asterias has indirect development. It means it is having a larval stage. So adult starfish, the gametes or the gonads of these adult starfish go into fertilization. The fertilization is external and it gives rise to blastula, then it gives rise to bipinnaria larva. The name of the larva in echinoderms is bipinnaria larva. Then that larva developed into again a small new offspring starfish by metamorphosis. Cross section through the body wall can also be seen. From outer to inner, the cross section shows that the outer layer is spine, then comes epidermis, then comes dermis, then followed by calcareous ossicle, tube foot or podia, coelomic cavity and digestive system. Important examples class-wise are Asteroidea, it has asterias, that is starfish, Echinoidea, echinus, sea urchin, and Crinoidea, antedon, sea lily, and Holothuroidea, Cucumeria, sea cucumber, and Ophioidea, that is brittle star. Aristotle's lantern in sea urchin, Echinoidea. So this Aristotle lantern is one of the very important example. As you can see that complex jaw apparatus used for scraping and grinding food, composed of 5 calcified teeth, is very important, and there is a basal piece there. This is present in the sea urchin in the class Echinoidea, and that is why it is called as Aristotle's lantern. The key points you have to remember is endoskeleton is of calcareous ossicles or spines. Water vascular system, a unique feature of the echinoderms. Adults are radially symmetrical, that is pentamerous radial symmetry, and bilateral larvae, and exclusively marine. Excretory system is absent. Tube feet or podia with suckers help in locomotion and food capture. Dioecious external fertilization, indirect development can be seen. That is all about phylum Echinodermata. You have to stop the video at this slide and revise this phylum multiple times. Moving on, phylum Hemichordata. Let's see. The silent features of the Hemichordata are organ system level of organization, bilateral symmetry, true coelom is present, digestive system is complete, circulatory system is open type. Distinctive features: worm-like marine animals, body divided into proboscis, collar and trunk, stomochord present in the collar region, rudimentary structure similar to notochord but not a true notochord, that is called as stomochord, hence it is called as Hemichord. hemichordata. They are not chordates basically. Respiration is through gills and excretory organ is proboscis gland. Reproduction is they are dioecious. External fertilization happens and indirect development with a larval stage. Examples are Belanoglossus and Saccoglossus. Let's see the external morphology. Example Belanoglossus. The external morphology shows proboscis being a preoral lobe, then there is a mouth in the ventral part, and there is a collar, and there is a trunk, and at the end there is an anus in the terminal. Body unsegmented, soft and warm like. Body is divided into three regions: proboscis, collar, and trunk. Internal organization can be seen in the transverse section. You can see the stomachord also. It is a rudimentary structure. It is a solid rod present in the collar region, dorsal to the gut. It is not a true notochord. The digestive system is complete. Circulatory system is open type, and respiratory system is through gill slits in pharynx, and excretory system actually is through proboscis gland called as glomerulus, acts as the excretory organ. Examples are Belanoglossus, commonly called as acorn worm. So wherever we use the word worms, either it is flatworms or roundworms. After that one, the use of worms was very little. In the hemichordata, we are using the word worm, but they are not true worms. They are acorn worms. They are hemichordates. Then comes the Saccoglossus, commonly called pterobranch. Proboscis not distinct, collar large, and trunk is short. This are the examples of hemichordata. Stop the video here and study all the other aspect. You have to remember especially the larva. The larva of the hemichordata is Tornaria larva, and study the previous year questions also. Moving on, phylum chordata. The phylum chordata, fundamental chordate characters present at some stage of the life, not always on the all the stages. One is presence of notochord. What is a notochord? It is a flexible supporting rod present at some stage. Second important fundamental character of a chordate is dorsal hollow nerve cord. Nerve cord lies dorsally and is hollow. The third important character is paired pharyngeal gill slits, open into pharynx, used for respiration in aquatic forms, and post-anal tail present beyond the anus, closed circulatory system, blood circulation in the blood vessels, and ventral heart lies ventrally to the gut. The chordate body plan can be seen here. Notochord lies between the nerve cord dorsal and the gut ventral. Pharyngeal gill slits open into the pharynx, and post-anal tail extends posteriorly. The subphyla of the phylum chordata are Urochordata, also called as Tunicata, Cephalochordata, and Vertebrata, also called as Craniata. Let's see what do you mean by Urochordata. Notochord present only in the larval tail. Adults are sessile, aquatic, enclosed in a tunic. Pharyngeal basket with numerous gill slits can be seen. Examples of Urochordata or Tunicata are Acidia, Salpa, and Doliolum. You can see the larva tadpole part here. Now comes the Cephalochordata. Notochord extends from head to tail region and persists throughout life. They are marine burrowing forms. Pharyngeal gill slits are present in numerous numbers. Example Branchiostoma, Amphioxus, and Lancelet. You can see the diagram here. Now comes the vertebrata craniata. Notochord replaced by cartilaginous or bony vertebral column in adults. Well developed brain and sense organs can be seen. Paired appendages like fins or limbs. Ventral muscular heart kidney is present. Example Petromyzon lamprey, Scoliodon shark, and Clarias catfish, rana frog, pigeon bird, and human all come under the vertebrata, also called as craniata. Comparison of the three subphyla, you have to do that one. You have to remember the examples and the images of the Urochordata, Cephalochordata, and Vertebrata, and integrate all these content with the previous year question. Stop the video here, study the whole slide carefully, and revise multiple times. Classes of the vertebrata. Subphylum vertebrata has many classes. Let's see first of all the silent features. The silent features of the vertebrata are jawless vertebrates, agnatha. They are marine but migrate to fresh water for spawning. Spawning is releasing of the offsprings and male gametes. It's part of reproduction. Elongated eel-like 6 to 15 pairs of gill slits. Circular, sucking without jaws. Mouth is present. Skin is without scales, paired fins are absent, skeleton is cartilaginous, cranium and vertebral column. Mode of life is ectoparasites on fishes. The reproduction, they are marine but migrate to fresh water for spawning. After spawning, adults die, larvae amniocetes metamorphose and return to ocean. Example are petromyzon and myxin. It is very important. Petromyzon is actually a neat favorite topic. So let's see the general structure of the class Cyclostomata, also called as Eknata, in which you can see the mouth is circular. There are 7 pairs of gill slits openings, no paired fins. There is one caudal fin, one dorsal fin. The circular sucking mouth can be seen here, and that is important character of these Petromyzon. You can see that the larva is actually amniocetes of the Petromyzon. The two living examples are Petromyzon lamprey and myxin hagfish. Common name of the Petromyzon is lamprey. Ectoparasite on fishes, has 7 pairs of gill slits, marine but breeds in fresh water. The common name of the myxin is hagfish, deep sea marine scavenger. No true vertebrae. Vertebral column is cartilaginous rod. The comparison between Cyclostomata and other vertebrates you have to do. Stop the video here and study the previous year questions. Now comes the class Chondrichthyes, cartilaginous fishes. The generalized characters of the cartilaginous fishes, that is Chondrichthyes, is habitat is marine streamlined body, endoskeleton is cartilaginous, notochord persistent throughout life, mouth is ventral, gill slits are 5 to 7 pairs separate without operculum, no gill cover, skin is tough with placoid scales, dermal tentacles, teeth are modified placoid scales backwardly directed, air bladder is absent, must swim constantly to avoid sinking, and heart is two-chambered, one auricle and one ventricle. Reproduction is internal fertilization. Many are viviparous. Males have clappers on the pelvic fins that acts as penis. And special features, some have electric organs like torpedo and some have poisonous sting like trigon. You can see the diagram there. The skin and teeth, the placoid scales can also be seen. The placoid scales are tooth-like structures. You can see the internal features, you can see the male reproductive structure, the special features of the torpedo and the poison sting, that is trigon, can also be seen here. The important examples are Scolidion, dogfish, Pristis, sawfish, uh, Carcharodon, great white shark, trigon, stingray, and torpedo electric ray. Stop the video here and study the whole slide along with integrating it with the previous year questions. Now comes the class Osteichthyes, called as bony fishes. The silent features of the class Osteichthyes bony fishes, they are marine and freshwater streamlined body. The endoskeleton is bony, mouth has terminal, gills are 4 pairs covered by operculum on each side, skin is covered with cycloid or ctenoid scales, air bladder is present, regulates the buoyancy, heart is two-chambered, reproduction is external fertilization, mostly oviparous, direct development, no larval stage. Examples are, you can see examples here in the downside. There are many examples there. Okay, scales in the Osteichthyes, one type is cycloid scales and the other is ctenoid scales. The air bladder can also be seen here, that is what makes them swim. You can see there is Exocetus, marine flying fish, Hippocampus, seahorse, also marine, and the freshwater are Labio rohita, rohu, and the freshwater Katla, and there is Clarius batrachus, magur, and the aquarium fishes like Betta splendens and Pterophyllum scalare. Stop the video and study the previous year questions given here. Now comes the class Amphibia. You know already all about Amphibia. Let's only study the important characteristic features and some examples. The habitat can live in aquatic and terrestrial habitats but need water for reproduction. Body is divisible into head and trunk, tail present in some example salamanders. Skin is moist without scales, rich in glands. Tympanum represents ear, external ear absent. Respiration is through gills in larvae, lungs in adults, also through skin, that is cutaneous respiration. Heart is 3 chamber, 2 auricles and 1 ventricle. Reproduction is external fertilization happens, oviparous, indirect development, tadpole, larvae. The respiration in the larvae is through gills. In adults, it is through lungs and also through skin, that is cutaneous respiration. Heart is 3 chambered. You have to remember this one. External features can also be seen. The life cycle of the frog is given here, Rana, and the examples of the amphibians are buffo, toad, rana, the frog, hyla, the tree frog, salamander, and ichthyophis, the limbless amphibian. These are the examples. Moving on, class Reptilia. Mostly they are terrestrial and some are semi-aquatic. Body covering has dry cornified skin covered with epidermal scales or scutes. The external opening, ear opening is absent. Tympanum represents ear. Heart is 3 chambered, 2 auricles and 1 incompletely divided ventricle, except crocodiles which have 4 chambered heart. Respiration, lungs only. Mode of locomotion, creeping and crawling mostly are terrestrial. Reproduction is internal fertilization, oviparous, egg-laying animals, and direct development. There is no larval stage. You can see in the diagram there is a tympanum, eardrum, external ear. You can see the heart is 3 chambered. The third chamber is incompletely divided chamber. In when it comes to crocodile, the heart is 4 chambered. Example: turtles and tortoises like Chilon and Testudo. The lizards are chameleon and calotis, and the crocodiles are crocodilus and alligator. Other lizards are hemidactylus, and the snakes like non-poisonous and poisonous can also be a part of reptilia. Example: Naja is cobra, Bangaras is kraken, Vipera is viper. Reptiles are ectothermic, cold-blooded vertebrates with dry, scaly skin that prevents water loss and helps in adaptation to terrestrial life. Class Aves. The general characters of the birds are also very important, and mostly everybody knows this one. Only specific named structures you have to remember, and you have to remember the names of the scientific names of the birds. So better stop the video here and study the whole slides on your own and integrate all the topics with the previous year question. Class Mammalia. Mostly all the topics when it comes to Mammalia are also known to every biology students. I need not to explain in detail. You just have to stop the slide here and study the whole slide in detail and integrate the slide points with the previous year questions. These are some of the corner concepts. It appears to be less important, but mostly direct questions are coming from these 27 things. Stop the video and study each and every question. Very, very important. These are the predicted questions for future NEET. Stop the video and study all the questions and also pen down on your notes. These are a few of the mnemonics to remember some of the topics which are usually volatile in nature. Like most commonly students can forget these things, so I have given those as mnemonics, memory tricks. These are frequently confused terms, so directly like term wise, definition wise questions can also come, so you should not forget these terms. Stop the video and study all these terms in details. This is a quick revision checklist from all the three, like biological classification, plant kingdom and animal kingdom, last 30 minutes before the examination. Common examiner traps also I have given you. Study all these 20 traps and see how he is preparing a trap and how to come out of the trap and reach the correct answer. So stop the video and study all the common examiner traps. And this is all about the previous year pattern analysis. So this completes our biological classification, plant kingdom and animal kingdom video. This video is very important, and I hope I'll find you in the another video. Stay tuned, do like the video and subscribe to our channel. If you have any doubts and queries and do you need any help, you can write in the comment section. Thank you.